Good morning, everyone. I'd like to welcome you to this FDA-sponsored workshop on the use of biomarkers to diagnose and assess treatment response in trials of non-alcoholic steatohepatitis. In my talk this morning, I will discuss how FDA approaches biomarkers for use as tools in drug development. Subsequent talks will cover specific applications for NASH. Next slide. Next slide. I'll begin by talking about the different types of biomarkers, then go on to talk about the evidentiary requirements for validation of biomarkers depends on the biomarker type and in particular on the context of use. Next, let's begin with the definition of a biomarker. Biomarker is a defined characteristic measured as an indicator of normal biologic responses, normal biologic processes, pathogenic processes, or responses to an exposure or intervention, including therapeutic interventions. Molecular, histologic, radiographic, or physiologic characteristics are different types of biomarkers. Next. The FDA worked with colleagues at the NIH to develop a categorization of biomarkers and the way that they're used in drug development. And this is called the BEST resource. Next. The BEST resource categorizes biomarkers into different categories. The ones shown at the top of the slide are biomarkers that are measures of disease presence and status, and the ones lower down are biomarkers that measure aspects of response to treatment. Next, an example would be a diagnostic biomarker. In the field of NASH, a diagnostic biomarker would be a liver histopathology to diagnose NASH. Next, a prognostic biomarker is a biomarker that's associated with prognosis of a disease. So, for example, elastography or blood biomarkers could be used to identify patients more likely to progress. Next, a monitoring biomarker is a biomarker that assesses some aspect of a disease or condition that can be evaluated repeatedly over time. Next, turning to the measures of response, a pharmacodynamic or response biomarker is one that changes in response to treatment. A particular type of pharmacodynamic biomarker is a surrogate endpoint biomarker. This is one that is expected to predict clinical outcomes later on. An example would be liver histopathology in NASH, which is accepted as a reasonably likely surrogate endpoint in NASH clinical trials. Next. Very important to consider the context of use. And for this, we look at both the BEST biomarker category and how the biomarker will impact clinical development of the product. So, for example, some biomarkers are used to enrich clinical trials in patients more likely to develop an endpoint, and this can be used to make clinical trials faster or smaller. Other biomarkers are used for dose selection, and other biomarkers, as you'll be hearing a lot about in subsequent talks, are used as surrogate endpoint biomarkers. Next. The specific context of use determines the level of evidence that's required to validate a biomarker. And we look at analytic validation and clinical validation separately. Analytical validation assesses the sensitivity of the biomarker, its specificity, its reliability, and its accuracy. Clinical validation measures how well the biomarker is associated with a particular clinical concept of interest. And I'd like to call your attention to one important consideration for clinical validation, which is benefit risk. And this benefit risk assessment is different from the benefit risk for a drug. In speaking about biomarkers, the benefit of a biomarker is that it can allow clinical trials to be accomplished more efficiently. And the risk of using a biomarker in drug development is that if it doesn't measure what it purports to measure, it can potentially harm patients. So, for example, if a surrogate endpoint biomarker actually does not predict long-term clinical outcomes, a drug could be approved that actually does not benefit patients. And then you could have safety issues without a benefit. Next. Biomarkers can be incorporated in drug development programs in a number of different ways. They can be incorporated as part of the drug approval process. So a pharmaceutical company sponsor would propose the biomarker to a review division, and the review division would provide feedback on incorporation of the biomarker in the drug development program. Another way a biomarker can be incorporated in drug development is through scientific community consensus. And finally, there's a program at the FDA, the Biomarker Qualification Program, where biomarkers can be qualified, and then they can be used in any drug development program for that particular context of use. Next. I want to emphasize that these three different pathways to incorporating biomarkers in drug development are not at all independent. Actually, they interact in very important ways. One important example is that biomarkers can sometimes be qualified in the biomarker qualification program for one context of use, for example, as a prognostic biomarker or as a monitoring biomarker, which requires a lower level of evidence than what's required for a surrogate endpoint biomarker. Then that biomarker can be incorporated into clinical trials prospectively by pharmaceutical company sponsors, deriving data that would support its use as a reasonably likely surrogate or as a surrogate endpoint biomarker. Next. Turning to the biomarker qualification process, this process begins with the requester submitting a letter of intent saying how they're proposing to develop their biomarker. Then, when that's accepted, the requester will submit a qualification plan specifying how they plan to qualify their biomarker and what the statistical analytic plan will be. Once that's accepted, the requester will carry out the qualification plan and submit the data to the FDA. If the data support use of the biomarker in that particular context of use, the biomarker will then be qualified, and as mentioned before, it can then be used in any drug development program for that specified context of use. Next slide. So finally, I'd like to provide these references that will help you access the resources that I mentioned earlier in my talk. And with that, I thank you, and I will turn it over to Dr. Stein, who will speak more about surrogate endpoint biomarkers.
Great, Jeff. Thank you very much. And certainly a pleasure to be here today. If I could have the first slide. Great. So what I'm going to try to do is pick up, in a sense, on the theme that Dr. Siegel has already started on talking about biomarkers. And I'm going to focus more on surrogate biomarkers that are functioning as surrogates, although not all surrogates have to be biomarkers, as I'll come back to. And surrogates can be used for important regulatory decisions, namely the ability to approve a new drug or to approve a new use of a drug. Go to the next slide, please. And I do want to focus on two particular uses of surrogates, or two types of surrogates, I should say, a validated surrogate and a reasonably likely surrogate. But to put that in the context, I think it's important to start by focusing on what FDA typically uses or expects to see to approve a drug, which is that we expect that there be substantial evidence of effectiveness, which is a body of information, typically from two adequate, well-controlled trials or an adequate, well-controlled trial and confirmatory evidence. But it focuses on demonstrating that the drug provides a meaningful benefit to patients. And we refer to that as how patients feel, function, or survive. That's the clinical endpoints that we would typically use to as a basis for an approval of a drug. We also recognize that there are times when that can be very challenging. For example, a disease that has a very low event rate, or a rarer disease, which is slowly progressive. So there are circumstances where the ability to do a trial using a clinical endpoint is limited. And in those circumstances, a surrogate endpoint potentially can be used. Now, a validated surrogate endpoint can be used to support traditional approval. You're familiar with these endpoints, such as LDL cholesterol and blood pressure, and HbA1c for approvals in reduction in cardiovascular disease as a surrogate to improve blood pressure, to produce cardiovascular outcomes, and to improve microvascular outcomes for HbA1c lowering. Now, in contrast, another type of a surrogate is what we call the reasonably likely surrogate. And this is a surrogate endpoint where there is some residual uncertainty, but where there is a strong basis to consider that it will, in fact, predict clinical benefit. And this is the type of surrogate that is used to support an accelerated approval, where after the approval, the sponsor would be responsible for demonstrating and verifying the clinical benefit of the drug. And go to the next slide, please. Now, it's important to sort of point out a few principles about the use of surrogate endpoints, both the principles and limitations. First of all, I think it probably would go without saying that a clinical endpoint, one that provides a basis to see the drug provides improvement in how patients feel, function, or survive, is always preferable to the use of a surrogate. But as I said, there are circumstances where a surrogate is appropriate based upon the feasibility of a trial, the need for a very large and long trial, or a very long trial, such as in a rare disease, which is slowly progressive. A surrogate is not exactly the same thing as a biomarker. It certainly often are biomarkers, plasma or matrix analyte, an imaging endpoint, histologic measure, a physiologic measure, but it also can be a clinical observation which is not directly tied to clinical benefit. It is not a direct measure of how a patient feels, functions, or survives, but it's intended to reflect and predict clinical benefit as a way of measuring or predicting the outcome. Next slide. Now, as I said already, because it is not a direct measure, it makes the benefit risk assessment somewhat more challenging, because we have to translate this indirect measure, surrogate, to the quantum of clinical benefit that it provides. There's also typically less safety information. The trials that are used to support a surrogate typically are smaller and maybe shorter, so the extent of safety information is also lesser. That makes the benefit risk calculation somewhat more challenging. But I will say that when we are making a determination as to whether there is sufficient benefit and that the benefit exceeds the risk, we do have to be able to have some understanding from the surrogate how much benefit we might expect there to be. Now, I will also say that there's always more reassurance in approving a drug with a clinical endpoint, because we're directly seeing the benefit that the patient is potentially obtaining from that drug. And that's always a question with a surrogate. So there's always a residual uncertainty. Certainly more uncertainty is accepted with a reasonably likely surrogate than with a validated surrogate. Next slide. Now, I do want to just touch on the fact that there's a range of types of surrogates. We have surrogates that might reflect the cause of the disease, such as serology or drug levels for drug toxicity, a variety of other factors that reflect the cause of the disease. There are also surrogates that are reflecting the pathogenic sequence of events. So there are biomarkers that are along the pathogenic pathway of the disease. And then there are also surrogates that reflect the end organ damage, either structural or functional losses that are associated with the disease outcome. Next slide. This is a bit of a complicated slide, and I won't go through this in great detail, but just to say, again, causal biomarkers that measure the mechanism, the etiology of the disease, pathway or mediator biomarkers that mediate the mechanism of disease injury, and then the, if you will, sort of outcome endpoint surrogates that measure either the structural damage or the functional losses associated with the complications of the disease. And those are most proximate to the clinical endpoints. Next slide, please. Now, you may have seen this slide from publications from Tom Fleming, and I think this is a useful construct to think about some of the limitations of surrogate endpoints. On the top, I'm showing slight modification from their graphic. As I pointed out already, the biomarker might reflect the etiology of the disease, it might be along the causal pathway, or might reflect the clinical outcome. And in all of these situations, it might nicely measure the effect that the drug has in improving the outcome of the disease. Next slide. Of course, the problem is that if you have a biomarker that is not on the causal pathway, it may be modified by the drug, but it may have limited impact on the outcome, or the drug might affect one of several different pathways, but only one pathway, and so not correlate terribly well with the clinical outcome. That makes the biomarker less clearly ability to predict the outcome of the disease with drug treatment. Next slide. And we also have the potential circumstance. Next slide, please. Thank you. You also have the potential circumstance where the drug may be on may modify a biomarker that's on the causal pathway, but it also may impose a toxicity, or the toxicity may actually harm the outcome. And so the biomarker may reflect some of the effect of the drug along the pathway of disease, but not all of the effects the drug has on either the safety or even on the outcome that the drug provides. And in which case, the biomarker is not a particularly good reflection. The surrogate is not a good reflection of the effect that the drug has on the outcome. I'll come back to this point with some examples. Next slide. Next slide. So, of course, I mentioned LDL cholesterol, a good example of a validated surrogate. And this was validated on the basis of a lot of underlying biology and understanding of the mechanisms of atherosclerosis and vascular injury. Of course, from extensive interventional clinical trials on the left with statins, and increasing data from non-statins that have a similar relationship, where the extent of reduction in LDL cholesterol predicts the extent of reduction in cardiovascular events. And this, along with very strong epidemiologic data, mechanistic data, all together certainly supports the use of LDL cholesterol as a validated surrogate. Next slide. On the other hand, we had the example of HDL cholesterol, where there is also a good epidemiologic relationship between increases in HDL cholesterol, higher, I should say, HDL cholesterol, and reductions in cardiovascular events. Next slide. On the other hand, when interventional trials were done here with a CETP inhibitor, there was no effect of raising HDL cholesterol and outcome. Now, is that because the hypothesis is wrong and HDL cholesterol increases don't lead to benefit, or is it because these drugs have other untoward effects that net do not lead to an outcome of improvement in cardiovascular events? Either of those explanations could be the case. But the point is that the biomarker or the surrogate, as predicted by epidemiologic data, did not turn out to be a surrogate in actually predicting outcome. Another example of this, a cautionary tale, if you will, next slide, is this next slide. Next slide. Sorry, a little bit of a build. Next slide. And one more. We're getting there. There we go. Thank you. The next example is one also well understood, well known, is the CAST trial. This is a trial that was evaluating whether reduction in premature ventricular contractions post-MI might lead to an improvement in outcome. And here they took patients who were post-MI, had PVCs. These were then, during the run-in phase, suppressed by one or two antiarrhythmic drugs. And then a randomized trial was done to see whether that suppression relative to placebo treatment would lead to an improvement in outcome. And the outcome was obviously the opposite, which is that the drug led to a worsening outcome relative to placebo. Why was that? Well, it wasn't necessarily that the hypothesis was wrong. It may be that reducing PVCs in some settings can improve outcome, but it may be that that hypothesis was in fact completely wrong. We don't really know the mechanism, whether it was an untoward effect of the drug in addition to the favorable effect, or that the hypothesis was entirely wrong. But in any case, it's another example of where a potential surrogate did not turn out to predict the outcome. And in fact, the opposite outcome occurred. Next slide. I want to spend just a few minutes talking about how one supports a surrogate. And I mentioned two types of surrogates: validated surrogates, which we can use to support traditional approval, and reasonably likely surrogate endpoints, which we can use to support accelerated approval. And I'd say that the nature of the data for these two types of surrogates are different. There's a wide range of data that can be used to support a surrogate. Randomized clinical trial with treatment group level data showing the relationship between the change in the surrogate and the improvement in the desired outcome. Individual patient level data where we're looking at the change that a particular patient has relative to an improvement in that patient's outcome. Observational data, epidemiologic and natural history data, mechanistic data, in vitro, in vivo, biologic data, pharmacodynamic studies in humans, genetic data, and translational animal models. A wide range of types of data can be used to support a surrogate. Next slide. Now, for a validated surrogate, typically we would be looking at treatment group level data, most commonly, as I showed you for LDL cholesterol, for example, as well as potentially individual patient level data. Doesn't always correlate the way treatment group level data does, but that can be very supportive, as well as observational data. And I don't mean to suggest that other mechanistic and biologic data isn't important, but the decision to utilize a validated surrogate more commonly rests on having clinical data, robust clinical data. Next slide. Now, in contrast, if you're looking at data that would support a reasonably likely surrogate, we also would like to see, and often do have, clinical data, whether it's patient individual level data or even treatment group level data. But very often the use of a validated of a reasonably likely surrogate may rest more on epidemiologic data, natural history data, mechanistic data, and a variety of other sources that lead to the biologic plausibility. And typically, again, the use of clinical data would be important where it's available and possible. There are rare diseases where that may not be entirely feasible to have extensive clinical data. Next slide. Now, in terms of getting to the acceptance of a surrogate, it's of course context dependent. In a rare disease, which is serious, where there's unmet need, obviously there's more flexibility in potentially accepting a surrogate, a reasonably likely surrogate endpoint. The impact of accepting the surrogate, what are the risks of the approval? Again, what do we understand about the drug? What is the safety of the drug? How is it administered? Those may also influence the flexibility one might have in accepting a surrogate endpoint, as well as the ability afterwards to demonstrate the drug and verify the benefit of the drug. We recognize that there's going to be different levels of evidence and types of evidence that are needed, as I said, for a validated surrogate compared to a reasonably likely surrogate. For a validated surrogate, again, treatment group level data from a randomized trial would generally be expected, whereas for a reasonably likely surrogate, we may be resting more on the biologic plausibility in concert with some epidemiologic data and some clinical data that may not be as extensive as we have for a validated surrogate. Typically, again, multiple sources of evidence would be needed in both of these situations. We typically wouldn't be simply using one type of source of data, but a range of data. And that leads us to thinking about this more as a convergence of evidence approach, putting together the information across clinical and preclinical information that allows the support for a conclusion that the surrogate either is expected to predict as a validated surrogate or is reasonably likely to predict the outcome. Next slide, please. And I'll end just by highlighting some of the challenges of developing both biomarkers, which then can become surrogates. Of course, there are many disease areas with unmet needs, and having insufficient drug development tools where a surrogate endpoint, particularly for those diseases that have a gradual, slow progression, can be challenging. Biomarker development is clearly a long and resource-intensive process, and so requires a lot of resources. And there are many stakeholders in this area with competing interests. There's often a lack of infrastructure to align the varying interests together. And so the challenge is prioritizing biomarker needs, focusing resources and integrating efforts across stakeholders. And so meetings of this type, where there's extensive discussion about the drug development tools needed to get to approvals, can be extremely important in getting people aligned and prioritizing the efforts to get the appropriate endpoints, both clinical and surrogate endpoints, together. So I appreciate your attention. And I think, am I turning it over to you, Dr. Prudis? Excellent.
Yes, I believe you are.
Thanks.
Thank you, Dr. Stein. That's great setup for my presentation. It's a pleasure to share with you a recent example in which a reduction in plaque on PET was established as a surrogate endpoint for accelerated approval. Next slide, in Alzheimer's disease. Next slide. I'd like to step back first and start with a brief note about the disease and therapeutic landscape at the time of our review. As we all know, the Alzheimer's is a devastating condition that leads to irreversible loss of memory and eventually loss of ability and functioning and death. The approved therapies at the time did not target the pathology of the disease, and the effects were transitory and small. Now, it's not controversial to state that Alzheimer's disease is a serious condition, but it's important to recognize what that implies. And we heard from many patients who are willing to accept a trade-off for some uncertainty about the clinical benefit for earlier access to a potentially effective drug. And next slide. So this slide shows the two pathological hallmarks of Alzheimer's disease, beta amyloid plaques and tau tangles. Now, if you focus on the right half of the slide, you'll see the plaques outside of the cell depicted as the red clouds, and the tau tangles inside the cell depicted by the red lines. Plaques are made up of a peptide fragment called amyloid beta, which can aggregate into bigger species and eventually plaque, as demonstrated in the process on the left part of the slide. Some of the aggregated forms of beta amyloid can be toxic to neurons and synapses. So buildup of plaque happens prior to clinical symptoms and before the spread of tau to different regions of the brain. It appears that it's required, however, for tau to spread. The new generation of anti-amyloid therapies bind and remove the aggregated forms shown on this plot with the blue line. The effect can be observed with PET imaging as described on the subsequent slides. Okay, next slide. The use of tracers to bind and visualize brain amyloid plaques took off really in 2002 with the introduction of Pittsburgh Compound B, which is closely followed by other tracers which have received FDA approval to help in diagnostic evaluation in Alzheimer's disease. The image on the right shows a negative scan on the left and a positive scan on the right, with the regions of higher intensity due to amyloid plaque. Now, PET imaging has been useful to aid in understanding of the temporal and spatial evolution of brain plaque in Alzheimer's disease. The two key developments were critical in the path to surrogacy. Next slide, please. Those two are quantification and harmonization. First, the PET signal was quantified using a standard uptake value ratio, which reflects plaque levels in areas of brain expected to be affected by pathology compared to regions spared of such pathology. And these values are dependent on the tracer and the platform used. So the second step as a key development was the creation of the Centiloid scale to standardize the values across tracers and imaging platforms. The Centiloid scale is a continuous scale. It's anchored to a value 0 in healthy young subjects and 100 in patients with Alzheimer's disease. And several groups have proposed thresholds of Centiloids of about 20 to 30, which can distinguish a positive scan from a negative scan, as you will see in subsequent slides and plots. Next slide, please. At this point, I think it's important to step back for a moment and say a few words about the wealth of scientific knowledge about plaques and their role in the disease. As I mentioned, plaque is a defining pathological feature of the disease, and strong evidence for its role in the disease comes from genetic discoveries in which mutations causing early onset disease occur in a reaction that produces amyloid beta and causes an overproduction of the protein, including in individuals with Down syndrome. And also there are genetic mutations which result in a decrease of amyloid production and appear to protect from the disease. At the time, there were also plenty of natural history studies which demonstrated that an advanced degree of pathology was associated with short-term cognitive decline. Now, despite all the plausibility shown in this slide and the extensive body of scientific knowledge, there was still uncertainty such that we did not accept a biomarker of brain amyloid as a surrogate endpoint. So for example, we know that there are some individuals who have brain amyloid who don't show cognitive symptoms, and that cognitive decline is better aligned with the spread of tau than brain amyloid. So what changed? Go to the next slide. Well, what changed is that we finally saw results from clinical trials of the newer generation of therapies, which resulted in a large reduction of brain amyloid as measured by PET. So on the left plot here, I show an example from the phase 2 study of aducanumab with the baseline scans on the left and the end of treatment scans on the right. The top row shows placebo, there's no change. And the bottom three show ascending doses, which show a dose-dependent change on amyloid PET. Similar results are shown on the right for lecanemab with a baseline scan in the top row and a post-baseline, post-treatment scan on the bottom, showing reduction of brain amyloid PET by PET imaging. Now, if you followed the public commentary 3 years ago, you might have heard that there are many previous drugs that reduced amyloid, but they always failed in clinical trials. And that simply is not the case. Previous therapies either did not target brain amyloid plaques or had no or very little effect on PET imaging. The development of the newer generation of therapies learned from the failures in the past. Go to the next slide. We'll take a look at the quantification of the reductions with a Centiloid scale. So the plot here on the left is a cross-study comparison showing key data from 3 drugs, which we had data available at the time of their review. The y-axis shows the brain amyloid level with the Centiloid scale, and the x-axis shows the time in weeks over the span of 1.5 years. The dotted line approximates the threshold between a positive scan and a negative scan. The gray solid line is a representative example of the time course of placebo arms, and the 3 color lines show results following treatment in 3 key studies for 3 drugs, which we had data for. Now, first, it's important to note that the placebo line does not change over time, which means brain amyloid does not go away in this population. The 3 colored lines show the time-dependent reduction during the course of treatment. By the end of the treatment period, the mean Centiloid value in the treatment arms is at or below the threshold of positivity. So these results show a clear, large effect due to treatment. So we finally saw a strong effect on PET imaging, but what about for clinical endpoints? Next slide, please. So now let's consider very briefly the effect on clinical endpoints that was observed in studies for these 3 therapies that I showed in the previous slide. There's not enough time to go into detail of all the data, but the key point is that when viewed in isolation, the available data for each program, some of which were still early in their development, were not sufficient to provide substantial evidence of a clinical effect. But when viewed in their entirety, the pattern of results was very clear and provided strong evidence of the reasonable likelihood of clinical benefit. So I'll go through pretty quickly the 3 programs. On the left, the aducanumab program had 1 large study, which demonstrated reduced clinical decline on the primary endpoint and all 3 secondary endpoints, but a second study of similar design was negative on the primary endpoint. A smaller dose-finding study had results on clinical endpoints that were similar to those findings on the positive study. In the middle, there's a lecanemab phase 2 study, which did not meet the primary objective, but the results for secondary endpoints at a later time point showed less decline from baseline in the high-dose treatment groups. And finally, on the right, the donanemab phase 2 study was positive on the primary endpoint with positive trends on secondary clinical endpoints. Importantly, across programs, there were some analyses that demonstrated a strong relationship between the reduction in amyloid plaques and reduced clinical decline at the group level, such that a reduction of brain amyloid to levels associated with a negative scan were associated with a reduction in decline across clinical endpoints by 20 to 40%. Next slide. So in summary, our conclusion was grounded in the basic biological and epidemiological information supporting the evidence of brain amyloid plaques as very important in a disease. Second, we observed a large dose and time-dependent reduction in brain amyloid as measured by plaques to levels consistent with a negative scan. At the same time, we had data from phase 2 studies that were coming in at the time supporting the relationship between the reduction in brain amyloid and reduced clinical decline. Next slide. So I'd like to leave you with a few key takeaways, which are very important for our decision-making. First, there's a rich scientific information, including natural history, biomarkers, and longitudinal PET data to support our review of the evidence. One helpful advancement was the harmonization of the measurement of the biomarker to enable cross-study comparison. It's also very important for us to understand the mechanism of action of the drugs and to be aware that not all anti-amyloid drugs are the same or produce the same biomarker effects. So it's important to be careful in how we assess the biomarker effects that we see in trials. And our experience was special in a way that some of the clinical information became available during our review, and it was critical for us to be ready to react in real time to new information as it came out. And finally, as I mentioned at the beginning, it is imperative to take into the patient experience into account. Now, as I said, it's important that our knowledge evolves as more trial data becomes available. So we go to the next slide. This slide shows the results of key studies, some of which have become available since our conclusion regarding surrogacy of amyloid PET, including results of large phase 3 trials. On this figure, each row is a study arm. The blue arrow begins at the mean baseline amyloid PET level in the treatment arm, and the arrow ends at the brain amyloid level at the end of the study. For the bottom rows, the bottom 5 rows, there was a large reduction of amyloid in which mean post-treatment values were at or below the threshold of positivity. These treatment arms were also associated with a 20 to 40% reduction across all clinical endpoints, as shown in the dark green squares. In treatment arms that had a reduction of brain amyloid, but not at levels reaching the threshold, trends were observed on clinical endpoints in favor of treatment, but not reaching significance. So these results confirm our conclusion and deepen our understanding of the relationship between brain amyloid and clinical benefit. Thank you. I'm going to turn over to Dr. Chang now to provide a different example.
Thank you, Dr. Curtis. If I could have my slide up. Next slide, please. Good morning. I am Christina Chang, Director of the Division of Urology, Obstetrics, and Gynecology in the Office of New Drugs in CDER. I have been asked by my colleagues in OND to talk about the experience with Makena, a drug that was approved under the Accelerated Approval Program in 2011. FDA withdrew Makena from the market this year due to lack of efficacy. For 12 years, Makena was the only drug approved to reduce the risk of preterm birth, despite having no demonstrated improvement in neonatal health. The story of Makena is a somber reminder that promising early data may not be substantiated later in larger confirmatory trials. Without demonstrated efficacy to balance risks, the profile of the drug is necessarily unfavorable. And without a positive benefit risk profile, there is no justification to keep the product on the market, even when there's high unmet need. Next slide, please. In this slide and the 2 following slides, I will provide general background on the condition and the regulatory framework on efficacy assessment. First, the condition of preterm birth, which remains a significant public health concern and one of the most pressing issues in modern obstetrics. Preterm birth is defined as birth prior to term or prior to 37 weeks gestation. And among the 4 million births each year in the United States, about 8% of singleton pregnancies and in babies being born early. Preterm birth is the leading cause of neonatal death and is a major cause of early childhood mortality and morbidity in the US. Babies born premature are at immediate risks for developing respiratory distress syndrome due to underdeveloped lungs, cerebral hemorrhage, and inflammation of the intestines that require surgery. Being born premature can also result in long-term physical and developmental challenges, such as cerebral palsy, debilitating hearing or visual problems, and learning disabilities. At this time, there is no approved therapy to reduce the risk of neonatal complications related to preterm birth. Next slide, please. The Food, Drug, and Cosmetic Act requires that all drugs demonstrate a substantial evidence of effectiveness before approval. There are two regulatory pathways to obtain FDA's approval for a drug. The first is the traditional approval, or full approval. The second is accelerated approval. In the traditional approval pathway, we rely on a validated clinical endpoint that directly measures how the patient in question, in our case, the neonate, feels, functions, or survives. In assessing therapies intended to treat the adverse outcomes of preterm birth, health outcomes in the neonate is the most relevant measure. When the therapy is supposed to improve health of the neonate, demonstrating actual improvement in neonatal health is necessary to establish the benefit of proposed treatment for preterm birth. With the accelerated approval pathway, though, we grant approval based on a surrogate endpoint that we believe to be reasonably likely to predict a clinical benefit. The advantage of an accelerated approval pathway lies in providing patients earlier access to promising therapy without waiting for a large pre-approval confirmatory trial. However, at the time when the accelerated approval is granted, there's less certainty in being able to translate the observed treatment effect into clinical benefit. And because of the uncertainty, a post-approval confirmatory trial is required to verify the clinical benefit. In the case of Makena, the gestational age at delivery was used as a proxy measure to assess the treatment effect. Next slide, please. This slide reinforces why gestational age at delivery is only a proxy measure and not a validated clinical endpoint. In pregnancy, a longer gestation generally reflects a healthier pregnancy and therefore healthier babies. However, we cannot assume the same for drug-induced prolongation of pregnancy, and certainly not in the case of Makena. The onset of labor is poorly understood. We don't know what causes the body to go into labor at term, much less preterm. And as I'm showing on the slide, the causes underlying preterm birth are multifactorial. These can include factors related to maternal health, such as maternal chronic diseases, subclinical infection, subclinical uteroplacental insufficiency, fetal reasons, or other reasons where the baby would be better off being delivered rather than remaining in utero. The mechanism of action of Makena is also unknown. So it is not clear if Makena was only merely keeping the uterus from going into labor despite an adverse intrauterine environment. If the onset of labor preterm is triggered by an unrecognized toxic uterine environment, we do not know whether artificially prolonging the pregnancy with drug therapy truly improves neonatal outcomes. Put another way, we don't have information demonstrating that neonatal outcomes from a drug-induced prolongation of pregnancy to, say, 32 weeks, 35 weeks, 37 weeks will be the same as those from a spontaneous preterm delivery at the same gestational age. It might be reasonably likely, but not certain. Next slide, please. So with these points for grounding, let me go into the Makena data in more detail. Makena, or hydroxyprogesterone caproate, is a synthetic progestin. The active ingredient HPC was first approved in 1956 for various gynecological indications. Smaller studies later suggested that HPC may prolong pregnancy in women who are at risk for recurrent preterm birth. In other words, those who already had an early delivery. In 2011, Makena received approval under the accelerated approval pathway to reduce the risk of preterm birth in women with a singleton pregnancy who have a history of singleton spontaneous preterm birth. The 2011 approval was based primarily on data from one randomized double-blind placebo-controlled trial comparing HPC to placebo. This trial was funded by the National Institute of Child Health and Human Development and conducted by the Maternal Fetal Medicine Unit, a network of academic hospitals in the US. Next slide, please. Results of this proof of concept trial were published in 2003 in the New England Journal of Medicine. Prior to the NDA submission in 2006, however, there was no engagement with the FDA. And as you can see, the primary endpoint evaluated was the proportion of women delivering prior to 37 weeks gestation. A treatment effect was shown for late preterm birth, meaning between 35 and 37 weeks gestation. Among women who were given Makena, 37% delivered before 37 weeks, while 55% of women given placebo did so. In other words, treatment with Makena reduced the incidence of preterm birth by 18%. At our request, the applicant also provided analyses that looked at, as secondary endpoints, the proportion of women delivering before 35 and 35 weeks and prior to 32 weeks. Next slide. Even though the trial collected information on pregnancy outcomes and clinical events in the neonates, the trial did not pre-specify in the analysis plan the assessment of neonatal outcomes. During the review, CDER requested these analyses be conducted. However, the neonatal outcomes, these endpoints can only be considered post-hoc analyses. And as shown here, treatment with Makena did not confer any survival benefit by reducing fetal or neonatal deaths. Next slide, please. Additionally, treatment with Makena failed to reduce neonatal morbidity, as shown by the results for the composite neonatal morbidity index here. So, because reduction in neonatal mortality and morbidity was not shown, Makena received accelerated approval and not traditional approval. Next slide. Prior to granting approval, though, we worked with the applicant to develop the study protocol for the confirmatory trial. And as in trial 002, trial 003 will be a randomized double-blind placebo-controlled trial, but almost 4 times the size of trial 002. Because the approval was based on an endpoint that was only reasonably likely to predict benefit, trial 003 was designed specifically to verify Makena's clinical benefit. To this end, trial 003 looked at 2 co-primary endpoints, a gestational age-related endpoint and a neonatal outcome endpoint. Both co-primary endpoints would need to be met to reach a conclusion of effectiveness. In trial 003, we asked for preterm birth less than 35 weeks because we considered it more likely to predict clinical benefit than preterm birth less than 37 weeks. We anticipated that recruitment would be difficult and made sure that at least 10% of the planned study population had been recruited from US and Canada before granting approval. As Makena became the standard of care in the US, recruitment outside the US indeed became necessary. We did not object to opening up sites outside the US because global clinical development programs are the norm in drug development. There's no biologically plausible reason to expect women at risk for recurrent preterm birth outside the US would respond to a progestin differently than US women. Next slide, please. Despite the planning, trial 003 took 10 years to complete. Results from trial 003 became available in 2019 and were disappointing. Not only did treatment with Makena fail to reduce neonatal mortality and morbidity, it also failed to reduce preterm delivery prior to 35 weeks gestational age. Also, there was no difference between Makena and placebo in the secondary endpoints of delivery before 32 or 37 weeks. So, trial 003 failed to verify clinical benefit, and importantly, also failed to substantiate the drug effect on the surrogate endpoint. Next slide, please. After receiving these results, CDER convened an advisory committee meeting in 2019 to seek input on the path forward. All 16 of the advisory committee panel members concluded that findings from trial 003 failed to verify the anticipated clinical benefit of Makena on neonatal outcomes. Most of the panel members also concluded that there was no substantial evidence of effectiveness. A small majority recommended that FDA withdraw Makena from the market. So in October 2020, CDER formally proposed to rescind the approval for Makena. And per the FDA and C Act, CDER also provided the applicant with the opportunity for a public hearing, which the applicant requested. Next slide. The 3-day administrative hearing took place in October 2022. CDER and the applicant both made presentations, followed by an opportunity for questions by the other party, the advisory committee members, and the presiding officer of the hearing. On the last day, after each party gave closing presentations, the AC discussed and voted on a series of questions. On the first question, do the findings from trial 003 verify the clinical benefit of Makena on neonatal morbidity and mortality from complications of preterm birth? All 15 members voted no and agreed that trial 003 did not verify the clinical benefit of Makena. On the second question, does the available evidence demonstrate that Makena is effective for its approved indication of reducing the risk of preterm birth in women with a singleton pregnancy who have a history of singleton spontaneous preterm birth? 13 advisory committee members voted no, 1 member voted yes, and 1 member abstained. During the discussion, most members concluded that with the 2 studies taken together, Makena's effectiveness had not been shown for the labeled indication. On the last question, should FDA allow Makena to remain on the market while an appropriate confirmatory study is designed and conducted? 14 advisory committee members voted no, and 1 member voted yes. The AC's discussions included whether Makena should remain on the market and whether facilitating another study was the reason to keep it on the market. The advisory committee also considered the benefit risk profile of Makena, factors related to unmet medical needs and equity, whether there was evidence in support of a narrower indication. Most AC members agreed that there was not sufficient evidence that Makena is effective in any population. Next slide. Following the hearing, the presiding officer, CBER's Deputy Center Director, issued her summary report in January this year. She concluded that Makena had not been shown to be effective, and its benefit risk profile was thus unfavorable. In April this year, after considering all the evidence and the records from the hearing, the FDA commissioner and the chief scientist agreed. They issued a joint decision ordering Makena to be withdrawn from the market. There have been many notable statements in the entire administrative records for Makena. And the quote I'm showing here from the final decision from the commissioner and the chief scientist is a very salient one because it underscores our careful considerations and why we took actions to uphold the regulatory integrity of the accelerated approval program. Next slide, please. So as we bring the Makena story to a close, I hope to leave you with a few points we learned from our experience. Surrogate endpoints could be used to support accelerated approval and bring needed therapy to patients sooner. However, because surrogate endpoints are proxy measures that are only reasonably likely to predict clinical benefit and have not been validated to correlate with clinical outcomes, confirmatory trials must be conducted to verify and describe the clinical benefit after approval. Enrollment challenges are bound to occur following accelerated approval, and these challenges can be mitigated by early planning and being realistic about longer trial duration. Finally, if confirmatory trial fail to verify clinical benefits of a drug approved under accelerated approval, FDA may pursue the steps to effectuate the withdrawal if certain conditions are met. And with that, I conclude my presentation. Thank you for your attention. I will now turn it over to Dr. George Meckert in DHN. Thank you.
Thank you, Dr. Chang. Good morning. My name is George McCarr. I'm the Acting Deputy Director for the Division of Hepatology and Nutrition. I have 2 goals over the next 15 to 20 minutes. The first is to provide an overview of the division's current perspectives on approval pathways for NASH and MASH drug development. The second is to place some of the concepts that have been covered by other speakers in this session in the context of considering NITs as surrogate endpoints in NASH drug development. Next slide. This is my conflict of interest and disclaimer statement. I have no financial disclosures. I have no conflicts of interest, and the views expressed are mine and do not necessarily represent an official FDA position. Next slide. I want to take a moment to comment on the new nomenclature for steatotic liver disease. Based on our understanding of the Multi-Society Delphi Consensus Statement on new nomenclature, and based on communication with the AASLD and other stakeholders, our understanding and our position is that MASH, or metabolic associated steatohepatitis, has the same histologic definition as what was previously defined as NASH. And so histologic enrollment criteria and histologic endpoints to support drug development for MASH are the same as those for NASH. NIT development for MASH would also be the same as for NASH. And so with that in mind, NASH and MASH may be used interchangeably in this presentation. And as a final note, other potential differences or other potential new areas of drug or NIT development with regard to the nomenclature changes, such as MET-ALD, are beyond the scope of this workshop. Next slide. Over the next 2 slides, I want to review some of the terminology and the concepts that were presented in Dr. Peter Stein's presentation and other presentations as well. First of is the concept of clinical outcome, which describes how a patient feels, functions, or survives. And clinical benefit is a positive, clinically meaningful effect of the drug intervention on that clinical outcome. In other words, how does the drug make a patient feel, function, or survive better than they did without the treatment? Traditional marketing approval requires this demonstration of clinical benefit, and accelerated approval requires demonstration of an effect on a surrogate endpoint that's reasonably likely to predict clinical benefit. And as we noted with the Makina example, we do require post-marketing confirmatory trials to verify clinical benefit. Next slide. So in terms of surrogate endpoints and approvals, we require a validated surrogate to be used to support traditional approval. And a validated surrogate requires both clear mechanistic rationale and clinical data that provide strong evidence that the effect on the surrogate reliably predicts a specific clinical benefit. Accelerated approval also requires a strong mechanistic rationale and/or epidemiologic rationale, but the clinical data may not be adequate to validate it as a surrogate endpoint, and that's why subsequent clinical trials to confirm clinical benefit are required. And the same applies to intermediate clinical endpoints. Finally, candidate surrogate endpoints have a role. Although they can't be used to support approval, oftentimes these may be used as primary endpoints in early proof of concept studies, and oftentimes they're captured in trials to develop the database that's required for them to ultimately, down the road, potentially consider them as either reasonably likely or validated surrogates. Next slide. So I want to review our currently accepted endpoints for NASH. On the left-hand side are endpoints to support accelerated approval, and the right-hand side, endpoints to support traditional approval. And as we know from our current draft guidances, we have accepted histologic endpoints to support accelerated approval, either resolution of steatohepatitis, improvement of liver fibrosis, or both resolution of steatohepatitis and improvement in fibrosis. And when assessing each individual endpoint, we do want to ensure that the other aspect does not worsen. So if we're resolving steatohepatitis, we also want to make sure liver fibrosis does not worsen. If we're improving liver fibrosis, we want to ensure that steatohepatitis does not worsen. On the right-hand side are traditional approval endpoints. The classic hepatic decompensation, transplant-free survival endpoints, I think are intuitive. And we have considered progression to cirrhosis to support traditional approval, with the understanding that clinical endpoints in chronic liver disease tend to develop in those who have progressed to cirrhosis. Next slide. I want to take a moment to acknowledge the concept of clinically significant portal hypertension. And although it's not clearly outlined and not really noted in our draft guidances, I do want to acknowledge that from an understanding from an FDA perspective, from a DHN perspective, we acknowledge that the rate of hepatic decompensation events in a cohort of compensated cirrhosis patients, or those who have advanced fibrosis who have not yet progressed to cirrhosis, can make it challenging to achieve an adequate number of clinical events. And just as we recognize that progression to cirrhosis is in some ways a clinical event, as it is a harbinger of negative outcomes, we also recognize that development of clinically significant portal hypertension is a precursor for most hepatic decompensation events. And again, although not included in our draft guidances, we have been open to including development of CSPH as a clinical endpoint. For example, as a component of a composite clinical endpoint. So for example, if you have a patient with a document of absence of esophageal varices at baseline, and during the treatment, they develop high-risk esophageal varices, we may consider that as a component of composite clinical endpoint. In addition, CSPH could be used when designing cirrhosis outcomes trials. Although not the focus of our workshop for today and tomorrow, it is something that may be considered when you're enrolling patients in a cirrhosis outcomes trial, in which you try to enrich your population for subjects who are more likely to develop clinical events in the context of the duration of a clinical trial. If you're going to enroll a mix of those with and without CSPH, it might make sense to consider stratifying enrollment between those two arms. Next slide. Here is one approach to drug approval with one trial population that can be used to support both accelerated and traditional approval. So often this is potentially one group of a trial population, for example, enrollment of 2,000 subjects ultimately, let's say, in a phase 3 trial, in which the first 1,000 or so are evaluated for NASH resolution, fibrosis regression, or both for accelerated approval. And that would be at the end of 1 to 2 years of therapy. You may continue to enroll patients until you are adequately powering your study for a clinical outcomes trial. And those patients continue to be treated in a blinded, placebo-controlled manner for several more years. We'd expect that in this type of a trial, in which at the time of enrollment, patients were non-cirrhotic, that the clinical benefit verification or clinical endpoints are probably going to be largely driven by progression to cirrhosis as the predominant clinical endpoint. Although, of course, capturing events such as clinically significant portal hypertension, hepatic decompensation events, and transplant-free survival are important as well. And so the idea here is that the phase 3 trial is initiated, your histologic endpoint is submitted for a surrogate endpoint, the trial is ongoing and largely enrolled, and then during your phase 4 confirmatory trial, you ultimately can submit that for clinical benefit verification. Next slide. An alternative to the sequential approach on the previous slide is to have 2 simultaneous trials. Trial 1 is for NASH patients with compensated cirrhosis that evaluates clinical outcomes, and trial 2 is for NASH with F2, F3 fibrosis that relies on the surrogate histologic endpoints. And in this approach, there's no need for a phase 4 confirmatory study, and you have an indication for the full spectrum of disease from NASH with fibrosis to cirrhosis. This approach does carry risks, however. The first risk is if your compensated cirrhosis trial fails to demonstrate clinical benefit. The second is if there's a safety concern that may arise that makes the benefit risk assessment different for non-cirrhotic versus a cirrhotic population. And in that setting, evaluation of outcomes in a non-cirrhotic population may be needed. Next slide. And that's why we strongly encourage continuing trial 1 until traditional approval, or in other words, until the compensated cirrhosis trial demonstrates clinical benefit, to ensure that at least one trial is capable of demonstrating clinical benefit. That way, if the compensated cirrhosis trial fails to demonstrate clinical benefit, you still have an ongoing trial in the non-cirrhotic population that can support clinical benefit in the non-cirrhotic population. Next slide. I want to take a moment to summarize again some of the concepts from the earlier presentations. To validate a surrogate endpoint requires a high bar, that you need both a comprehensive understanding of mechanistic rationale and a large body of clinical data. And it requires, therefore, a clear understanding of the causal pathway of disease, including how that causal pathway leads to clinical outcomes. You need to understand where the surrogate lies along that causal pathway, how the drug fits into the causal pathway, and whether there are off-target effects that are not captured by the surrogate endpoint. And although correlation between the surrogate and the clinical endpoints is necessary, it's not sufficient. You need to demonstrate that the full or at least most of the effect on the drug and the clinical endpoint is explained by the surrogate. Often meta-analyses across clinical trials of a drug class would demonstrate that the drug effects on a surrogate reliably predict the drug effects on the clinical endpoint. So the validity of the surrogate depends on the disease, the clinical endpoint of interest, and on the drug mechanism of action, as noted in the previous two presentations. Next slide. So for illustrative purposes, I want to, for a moment, compare chronic hepatitis C with NASH-MASH. SVR12, or sustained virologic response at 12 weeks, is considered a validated surrogate endpoint for directly acting antiviral therapies for Hep C. And as we've already reviewed, histologic endpoints are considered as reasonably likely surrogates for NASH and MASH. Why is SVR12 a validated surrogate? Mechanisms of disease are well defined. We have a single agent of disease. We understand that antiviral therapy clearly impacts the causal pathway of disease. We have well-validated methods of assessing the surrogate, meaning of measuring HCV RNA. We have robust data from multiple sources that viral clearance impact long-term clinical outcomes. And we have data for clinical benefit on liver outcomes demonstrated by viral clearance with different drug classes. In contrast, although chronic inflammation and fibrosis predict outcomes, and we see that not only in NASH and MASH, but in other chronic liver diseases, which is why we've accepted these as reasonably likely surrogates in support of NASH-MASH. The mechanisms by which MASH leads to chronic inflammation and fibrosis is complex. Drugs currently in development are unlikely to affect all the causal pathways of disease. There's an absence of empirical evidence or clinical data to support drug treatment can benefit NASH or MASH cirrhosis outcomes. And there's insufficient empirical evidence to identify a clinically meaningful change in a biomarker that clearly predicts clinical benefit. Next slide. I want to conclude with a few considerations of NITs or non-invasive tests as potential surrogates. A biomarker that predicts a reasonably likely surrogate or a validated surrogate is not necessarily in and of itself a reasonably likely surrogate or a validated surrogate. So when we use, for example, NITs to predict liver histology, that NIT in and of itself is not necessarily a reasonably likely surrogate. It can still be useful for patient selection and enrichment, especially when we're considering clinical outcomes trials. It can be used as a PD biomarker, an endpoint, an early proof of concept studies to gain initial evidence of effectiveness. But what we're really looking for are NITs that predict clinical outcomes that may eventually serve as reasonably likely or validated surrogates. Next slide. The majority of NITs that have been proposed as potential surrogates or that can be used as prognostic biomarkers do not necessarily differentiate the relative contributions of steatohepatitis versus fibrosis. And although some biomarkers do attempt to identify the presence of inflammation, their utility for use as a prognostic or a disease response biomarker is unknown. And to illustrate this, and again, not to pick on any specific NIT, but to illustrate this, I want to look at the concept of elastography. As we all understand, liver stiffness is contributed to or is a combination of both steatohepatitis or inflammation in general and fibrosis. in addition to extrahepatic etiologies, for example, right-sided heart failure, et cetera. And so we see this going back to the chronic hep C example. I'd like to sort of touch on this point, which is, as we all understand, if in our clinical practice we are using elastography and we're assessing liver stiffness as a patient with hepatitis C, we understand that the same level of liver stiffness in an untreated hepatitis C patient does not predict the same likelihood of advanced fibrosis as in a treated hepatitis C patient. And that's because the contribution of inflammation to liver stiffness has resolved in the patient who's achieved SVR. And so the question when we take this to NASH is, does a decrease, let's say in a 5 kilopascals on VCTE or a decrease in 1 kilopascal on MRE, does that predict the same amount or quantum of benefit regardless of the drug mechanism of action? For example, an anti-inflammatory or an anti-fibrotic or a drug combination that has effects on both. Next slide. A change in a biomarker that predicts outcomes, for example, an increase or a worsening in liver stiffness or ELF or some other circulating or imaging biomarker that predicts negative outcomes or harm, doesn't necessarily mean that a similar magnitude of change in the opposite direction in response to therapy predicts a similar magnitude of benefit on those outcomes. So to provide support for a biomarker as a surrogate, you also need to demonstrate that an improvement in the biomarker is associated with clinical benefit. And this was illustrated by Dr. Stein's presentation in his discussion of HDL, where we all understand that there's an association of cardiovascular risk with HDL levels in epidemiologic studies, but an increase in HDL with drug therapy did not necessarily translate into cardiovascular benefit. And we see this in other diseases as well. For example, CD4 counts in HIV, in which improved CD4 counts predict improved outcomes. But the primary pathway of treatment is really HIV viral suppression, not a direct effect on CD4 counts. Prospective trials in which biomarkers are captured at baseline and to clinical outcomes may provide the data to characterize what magnitude of change in a biomarker can predict a benefit on clinical outcomes. And I'll end here by proposing a hypothetical example of MR elastography, not to single out MRE, as we would ask the same set of questions of other imaging or circulating biomarkers. But what are some of the clinical validation questions that we would want to consider? First, we'd want to ask, what minimum level of change predicts clinical outcomes? What's a minimum level of change that predicts clinical benefit? So for a hypothetical example, let's say 1 kilopascal. Does a 1 kilopascal decrease on MRE provide the same benefit regardless of drug mechanism of action, anti-inflammatory, anti-fibrotic, or both? Does a 1 kPa decrease predict similar benefit within drugs of the same class or mechanism of action? Does a 1 kPa decrease on MRE predict a similar benefit regardless of stage of disease, age, sex, race, BMI, presence of comorbidities? And how long does a 1 kPa decrease need to be maintained to provide clinical benefit? So in summary, to validate a surrogate endpoint, we would require prospective data from multiple sources that demonstrate that a benefit on the surrogate does in fact predict a benefit on clinical outcomes. Thank you. I will turn it over now to Dr. Hager.
Hello, my name is Rebecca Hager, and I'm a statistical team leader at FDA, supporting the Division of Hepatology and Nutrition. My presentation will continue the discussion from previous speakers about biomarkers, surrogate endpoints, and advancing endpoint development. Here's my disclaimer that this presentation reflects my views and should not be construed to represent FDA's views or policies. Some of the material I present today is adapted from work by Lisa McShane at NCI, Laura Lee Johnson and Greg Levin at FDA, and the BEST resource. There are a few key steps when considering biomarkers and candidate surrogate endpoints, which previous presenters have also touched upon. First is clearly defining the intended use of the proposed biomarker, which I will refer to as the context of use, or COU. Validation is a process that is related to a specific intended purpose. A biomarker that has been determined as fit for purpose or validated for a non-regulatory use or a given regulatory purpose is not necessarily sufficient for another regulatory purpose. Therefore, you may see that a biomarker is referred to as validated in the literature, but that does not necessarily mean it is validated for every regulatory purpose. It is also important to understand the potential benefits and risks associated with the proposed context of use. For example, there is a lower risk of using a biomarker as a diagnostic or prognostic tool compared to relying on a biomarker to define an endpoint to support a regulatory decision. Last is collecting the right evidence to support the intended context of use. For example, this may include evidence from completed, randomized, controlled interventional trials that evaluate both the biomarker and clinical outcomes following treatment. Previous speakers have gone over the terminology for different types of outcomes and endpoints, and I will briefly review a few key terms. A clinical outcome is an outcome that describes or reflects how an individual feels, functions, or survives. A clinical benefit is a positive therapeutic effect on this outcome that is clinically meaningful. There are a range of different biomarker types, as previously described by Dr. Siegel, but I will mention two, which I will discuss further. A prognostic biomarker is used to identify the likelihood of a clinical event, disease recurrence, or progression in patients who have the disease or medical condition of interest. A predictive biomarker is used to identify individuals who are more likely than similar individuals without the biomarker to experience a favorable or unfavorable effect from exposure to a medical product. A surrogate endpoint is a marker that is thought to predict clinical benefit, but is not itself a measure of clinical benefit. Previous presenters have defined reasonably likely and validated surrogate endpoints and the approval pathways that each may support. There are a few common themes that often come up in discussions about biomarkers and surrogate endpoints that can lead to issues that Dr. Stein and others brought up. There can be confusion around definitions and levels of evidence needed to support the different contexts of use. There is not a one-size-fits-all approach across disease areas and indications, which may contribute to this confusion. One point of confusion is distinguishing a prognostic biomarker and a predictive biomarker, which I will give an example of next. Another issue is providing support for a potential surrogate endpoint based solely on correlations of outcomes. We know that correlation does not imply causation, and I will go through some examples on how this could go wrong when considering surrogate endpoints. First, I will show examples of prognostic and predictive biomarkers. This figure depicts an example from the BEST resource of a biomarker that is prognostic but is not predictive. This left panel depicts survival curves over time of patients on an experimental treatment. The black line indicates patients that are biomarker positive, and the lighter gray line indicates patients that are biomarker negative. We see that even though these patients are on the same treatment, this biomarker may be used to identify the patients who have a higher likelihood of surviving, which is a prognostic biomarker. Now looking at the right panel, there are added dotted lines for the patients on the control treatment. We see that there is no apparent difference between the survival of patients on the experimental and control treatments within the same biomarker status, so this would not be a predictive biomarker. This next figure depicts an example from the BEST resource of a biomarker that is both prognostic and predictive. The patients who are biomarker negative in gray do not show a treatment effect on survival, while the patients who are biomarker positive in black show a treatment effect by the separation between the dotted and solid black lines. Moving on to discussing potential surrogate endpoints, I will go through a few causal diagrams depicting hypothetical candidate surrogates and clinical outcomes. This figure depicts an ideal surrogate where the surrogate is directly on the causal pathway of the disease and the treatment effect on the surrogate captures the full effect on the clinical endpoint. This next figure depicts a surrogate that would be problematic. In this case, the disease affects both the surrogate and the clinical endpoint, and these two outcomes are correlated. However, the surrogate endpoint is not on the causal pathway to the clinical endpoint. Therefore, an intervention may have an effect on the surrogate endpoint that will not be reflected in the clinical endpoint. These types of surrogates may be useful for diagnosis or prognosis as they are related to the disease, but they are not sufficient as clinical trial endpoints to evaluate a treatment effect. There are more specific examples of these causal pathways in this paper by Drs. Fleming and Powers. Another important consideration when thinking of surrogate endpoints is to consider the mechanism of action of the treatment. This example is similar to one that Dr. Stein presented earlier. In this case, the surrogate endpoint is directly on the causal pathway to the clinical endpoint. However, the surrogate endpoint isn't fully capturing the effects of the treatment. Therefore, treatment may have unintended on or off-target harmful effects on the clinical endpoint, which would not be apparent when evaluating the surrogate endpoint. Not only do we not want to miss if there are potentially harmful effects of a treatment, but we also don't want to miss if a drug has a beneficial effect on the clinical endpoint that may not be apparent on the surrogate endpoint. This is why some trials save alpha for evaluating a final clinical endpoint, even if the reasonably likely surrogate endpoint fails at an interim analysis of the trial. There are no perfect surrogate endpoints, but there are different aspects to consider when proposing or evaluating a candidate surrogate endpoint. The previous examples of causal diagrams support the need for multiple sources of evidence for surrogate endpoints and to improve upon the evidence often presented in the literature to justify that biomarkers are surrogates. This slide is adapted from a presentation by Dr. Lisa McShane from NCI. Some questions to address to support use of a surrogate endpoint include whether there is a compelling case for the surrogate being on the direct causal pathway to the disease outcomes, whether there is compelling biological evidence for the surrogate that adds to the weight of the empirical evidence, whether there are other factors affecting the disease outcome, including off-target effects of the drug, the extent to which the magnitude of change in the surrogate endpoint explains the magnitude of change in the clinical outcomes, and the extent to which there is evidence across drug mechanisms or across different populations. I am borrowing this graphic from an FNIH presentation of Dr. McShane's because it helps walk through these elements. It is important to understand both the disease process and the relationship between a drug's effect and the disease process. Bottom up, working through biomarker discovery, usually we start by finding out there is a difference between non-diseased or normal and diseased. Then we look to see if there is correlation with the values from the biomarker measurement and disease severity. With both of those and biological reasoning, we have a candidate. Next, we need to know if the biomarker is prognostic of disease progression or severity. Remember, correlation ties to disease severity. It does not imply that a biomarker is predictive of clinical outcomes following treatment. The top band is where we can utilize double-blind, randomized therapeutic trials measuring clinical benefit endpoints and the candidate surrogate after delivery of treatment. The amount of change also matters. Ideally, patients who have high clinical benefit would have large effects on the biomarker. Similarly, patients with low clinical benefit would have small effects on the biomarker, and this would translate at the in-between points across the measurement space. Unfortunately, we cannot determine that a biomarker can serve as a validated surrogate without evidence on the clinical endpoint. As Dr. Stein mentioned, usually there are multiple trials and meta-analyses to make this assessment. To get here, we will almost always need randomized interventional clinical trials and not simply observational studies. We are lucky that in NASH there are many trials generating a lot of data. Hopefully there can be collaboration so the community can utilize trials in the relevant patient populations and the drug classes of interest, evaluating the appropriate endpoints, etc., to evaluate candidate surrogates. In conclusion, the level of evidence needed to support a biomarker or surrogate depends on the intended context of use. Prognostic is not sufficient for surrogacy. Completing trials to verify clinical benefit is not only required for traditional approval following accelerated approval, but could also support the use of biomarkers and surrogate endpoints in future trials. We recognize that there are many challenges in NASH clinical trials. However, premature adoption of poor surrogates could thwart efforts to answer questions about clinical benefit. We are hopeful that endpoint development will continue to advance as trials move forward in this space, and that is one of the reasons we are here today. Thank you.
Good morning. I'm Dr. Laura Lee Johnson, and I direct the Division of Biostatistics that supports CDER's Division of Hepatology and Nutrition. We have 5 speakers in this session on endpoints in clinical trials and 3 additional panelists with a late lunch break for those of us on the East Coast, so we'll be strict on time. I'll introduce all 5 speakers now so we can more quickly transition between the talks. Our first 2 speakers will focus on one-stage reversal of fibrosis. First, Dr. Don Rockey will give a hepatologist view. Dr. Rockey is a professor of medicine specializing in gastroenterology and hepatology at the Medical University of South Carolina, where he leads bench-to-clinic research. Next, Dr. David Kleiner from the National Cancer Institute at the NIH will provide the pathologist view. Dr. Kleiner's research in hepatic pathology is focused on liver cancer and chronic liver disease, with particular attention to assessing histological responses to therapy. After that, we'll turn to NASH resolution. Dr. Naga Chalasani, who is co-moderating this session with me, will provide the hepatologist view. Dr. Chalasani is a professor of gastroenterology and hepatology at the Indiana University School of Medicine. His research is focused on NAFLD and drug-induced liver injury. Our fourth speaker is Dr. Cynthia Bailing, who will provide the pathologist view. Dr. Bailing specializes in hepatobiliary and gastrointestinal pathology at the Pacific Rim Pathology Lab. Her research interests include histologic endpoints for the clinical trials and fatty liver disease. Our last speaker will be Dr. Theo Heller. Dr. Heller is the section chief at the NIH NIDDK Translational Hepatology section. His work focuses on rare liver diseases, effects of systemic diseases on the liver, and approaching common problems in liver disease in a multidisciplinary manner. After Dr. Heller, we'll take a short break, and then Naga and I will introduce our additional panelists and the start of the discussion panel. We encourage you to submit your questions throughout the session. Thank you in advance to all our speakers and panelists. Don, the floor is yours.
Okay, thank you. Good morning, everybody. If I could get my first slide. All right, so I'd like to start by thanking Doctors Mehta and Vanderpala for the kind invitation to present. I'll be talking about one-stage reversal of fibrosis. How do hepatologists view this change? Next slide, please. These are my disclosures. I don't accept any money myself. It all comes to the university, and I don't do stock or speakers bureaus. Next slide, please. This is an overview of what I'll be covering in the next 15 minutes. We'll talk about prognosis in fibrosis, pathogenesis of fibrosis. I'll spend a fair amount of time talking about fibrosis regression and reversal. And then I'll try to put some of the antifibrotic trial data in perspective and conclude by asking, what does this all mean? Next slide, please. So the first question, I think, for this morning is, why is liver fibrosis important? Next slide. And I believe the data are now relatively convincing. So I'm going to show you a couple of studies. This is a really an elegant study from an Alaskan cohort of patients with hepatitis C. who underwent a liver biopsy at baseline and had their histology staged. You can see there, this group used the Ishak staging system, which goes from 0 to 6. And then they simply followed patients over time until they developed a complication of liver disease or end-stage liver disease. And as you can see here, it's very clear that the more severe, the higher stage of fibrosis you have, the more likely you are to develop an event. Next slide, please. The data are analogous in NAFL, MASH, ASH, NASH, whatever we want to call this now. And you can see this is a Swedish study, again, biopsy at baseline, histology, stage from F0 to F4. And if you follow the lines, the more advanced the fibrosis, the greater the likelihood of death. Next slide, please. And the NASH CRN has published similar data. So shown on the left are mortality data, and on the right, hepatic decompensation data. And I think it's very clear here that you can see the more advanced the level of fibrosis, F3 and F4 in particular, the greater the likelihood of either mortality or a hepatic decompensating event. Next slide. And there are meta-analyses. There are data provided by meta-analyses as well. So this is a recent one of some 17,000 patients that examined all-cause mortality at 1, 5, and 10 years, which was increased significantly. F2 fibrosis was even associated with an increase in all-cause mortality. You can see in the graph over on the right side, green is no significant fibrosis, but other stages are clearly associated with increases in mortality. Now, this study, as many of the patients included in these studies, are limited by a lack of time to event information, and some of the sample sizes were small with regard to liver-related mortality. Next slide, please. So I think, and hopefully we will all agree that fibrosis itself has adverse clinical complications, and I think is an important clinical endpoint. Next slide. So let's talk a little bit about pathophysiology. You've heard some references this morning already to the importance of biology and mechanism and thinking about endpoints and surrogates. So in terms of fibrosis, fibrosis is essentially the body's wound healing response to injury. So in most parenchymal organs, there is injury to an epithelial cell, shown on the top here. Subsequently, there is an inflammation, which leads to activation of effector cells. These effector cells often undergo some sort of paracrine process with regard to inflammatory mediators, and as well may have autocrine effects that in which these cells are self-perpetuating effectors. The cell, as we all know in the liver, the effector cell, as we know in the liver, is the hepatic stellate cell. Next slide, please. Which undergoes a process of activation. And this cartoon simply depicts the fact that in the normal liver, the stellate cell is quiescent. After injury, typically an inflammatory response, there is activation, and the activation process is associated with a number of important phenotypes, prominent among which is fibrogenesis, which is depicted here by the blue strands of matrix. Next slide. So the take-home point here in terms of mechanism and pathobiology is that the biology here is dynamic, and it involves multiple elements, including cells. I've shown you a little bit about hepatic stellate cells, but also Kupffer cells, endothelial cells, immune cells, and others. There are multiple molecules involved. TGF-beta is the canonical fibrogenic cytokine, but there are many others involved, and there are many pathways involved as well. Matrix structure itself is important, and remember this: the matrix undergoes a dynamic process where there is both deposition and degradation. So this is not a static component at all. Next slide, please. So not only in the liver, but in other organs, heart and kidney, the evidence is that fibrosis is reversible. Next slide. So with regard to liver, what are the approaches to stop or reverse fibrosis? We could eliminate the inflammation when it is present. We might accelerate the reversion process. We could modify the cells in the liver, the stellate cell, endothelial cell, Kupffer cells, immune cells, others. We could stimulate extracellular matrix degradation. We might block in early inhibitor effector cell function, or we might degrade matrix directly. Next slide, please. So what is the evidence in human liver disease? So this is a table that highlights diseases in which fibrosis can be reduced by treating the underlying disorder. So the best evidence is in hepatitis B and hepatitis C, and I'll review some of those data shortly. I think all clinicians have experiential experience in alcoholic hepatitis, where alcohol cessation often leads to improvement and likely reversal of fibrosis. Data in NASH are evolving, and I think there's excellent evidence now with regard to weight loss, particularly in the bariatric surgery field, in that this leads to reversal of fibrosis. And then several other liver diseases are listed below. Next slide. So what about hepatitis B? This is a table, a figure that highlights the change in fibrosis over time. These are data from a randomized controlled study of tenofovir and adefovir for over 7 years. Almost 350 patients had paired baseline and week 240 liver biopsy and histology assessment. And again, this is the Ishak scoring system. You can see here that at baseline, a substantial proportion of patients had Ishak 5 and 6. Some patients had early stage fibrosis, but what is key here is that the proportion of patients with advanced fibrosis, Ishak 5 and 6, declined, while the number of patients with early stage fibrosis increased. And in fact, there were some patients whose fibrosis seemed to disappear entirely. Next slide. The data in hepatitis C are also remarkable. This is a study from a histomorphometric analysis of 38 consecutive Italian patients with cirrhosis, so Child-Pugh class A, who had SVR after PEG interferon and then had 4-year follow-ups. The investigators did picroserious red morphometric analysis of collagen, as well as immunohistochemistry to detect smooth muscle alpha actin, a marker of stellate cell activation. And they showed a median reduction of 72% of the area of fibrosis. And you can see in the graph on the right, on the y-axis is the area of fibrosis, and the x-axis is pre and post treatment. And you can see that most patients had a decline in the area of their fibrosis. Next slide. What about clinical outcomes? So this is a study that that I participated in with Andrew Moon and George Wanu of nearly 35,000 veterans with hepatitis C that was eradicated by DAA, but also included some patients who did not have an SVR. Patients were followed for a little over 3 years. And you can see here in the graph that that SVR was associated with a reduction in the risk of variceal bleeding in patients with or without cirrhosis. And this risk reduction was maintained in a variety of subgroups. So clearly eradication of hepatitis C and likely, although not proven in this study, reversal of hepatic fibrosis was associated with improved clinical outcome. Next slide, please. And hepatitis B, I suspect most of you are familiar with these data, which are now nearly 20 years old. So this is from the landmark study by Liao and colleagues, which randomized patients to lamivudine or placebo and showed that even as early as 12 to 18 months, suppression of hepatitis B in infected patients with advanced liver disease led to an improved clinical outcome. Next slide. So what about specific antifibrotic therapies in human liver disease? There have been a number of trials. This is a table that highlights and summarizes the data from many of these studies. And these trials are predicated on outstanding preclinical and mechanistic data. So that is to say that these compounds have clear effects in animal models on fibrosis and or hepatic stellate cells, and clearly demonstrate a benefit. Unfortunately, in humans, while most of these are safe or relatively safe, the efficacy is simply not there. The jury is still a little bit out on statins, and I'm not sure whether we will ever see a pure antifibrotic trial in statins, given their overall beneficial effects in other areas. Next slide, please. So the story in fibrosis trials to date is that nothing really has shown great effectiveness. And again, this is in pure antifibrotics, which raises the question, are we studying the right disease? Do we have the right trial design? Or perhaps some of these medications are simply ineffective. Now, a challenge, and we're going to talk about that next and throughout the rest of the day today and tomorrow, is that histology and fibrosis in particular has been a primary outcome in many of these studies. Next slide, please. But there are some questions about fibrosis as an outcome. And those include, first of all, we measure fibrosis in a linear fashion. So most studies are using an F0 to F4 staging system, which may or may not be appropriate. Fibrosis is almost certainly not linear. And therefore, I think this is an open question. There are differences in the way we look at fibrosis, and I look forward to hearing the comments by our pathologists. There are some septae are thin, some are medium sized, some are thick. And then, of course, another key issue is whether fibrosis reversal at 12 or 18 months or perhaps 24 months is truly realistic. Next slide, please. Now, this latter question has been addressed somewhat by the REGENERATE trial, the data of which, or a brief overview of which are shown here. So I suspect most of you are familiar with this trial. This is a double-blind, placebo-controlled, international, multi-center study of obeticholic acid in patients with NASH and advanced fibrosis. One of the primary outcomes was at least a one-stage improvement in fibrosis with no worsening of NASH. And the data are shown here in this table of some 900 patients, in which I think it's clear that fibrosis was improved in patients receiving the highest dose of obeticholic acid. Now, I'm not going to go into detail about the advisory panel meeting and their recommendations, but I would make the point here that the p-value here is highly significant, and I think that it's clear that fibrosis was improved and reversed in a proportion of patients in this trial. Next slide, please. So, based on the available data, it's my belief that a treatment that clearly halts fibrosis progression or reverses fibrosis will, in fact, ultimately lead to clinical outcomes. And I hope I've shown you enough data to convince you of that point. Thus, and this is my opinion, the question is not really whether a change in fibrosis is clinically meaningful or not. I think that it is. I think if we can halt fibrosis or reverse fibrosis, better yet, that is good. But I think the question really is whether it is possible to reliably measure a one-stage change in fibrosis. Next slide. Next. So I don't want to steal the thunder of our pathologists and our non-invasive liver disease assessment experts, but I will kind of frame the discussion a bit by saying that there are issues with both of these techniques which are used to assess fibrosis. So liver biopsy, is tried and true. We've been doing liver biopsy for 50, 60 years now, but patients don't like it, physicians don't like it, and it is associated with significant complications. Death occurs in a small proportion of patients. It's also very expensive and time consuming. So the non-invasive approach is gaining substantial notoriety and enthusiasm now, but it is not perfect either. And I think the critical question, at least in my mind, is that there are issues with the accuracy in the intermediate and early stages of fibrosis. So I look forward to hearing more from our experts about that. Next slide. So where do we stand? The take-home points from my talk are that fibrosis is associated with poor clinical outcomes, it is dynamic, and it is clearly reversible. To date, fibrosis reversal has been well established when the underlying disease is controlled and is associated with improved outcomes. Whether fibrosis can be treated independently of treating the underlying disease is an important but more difficult question. And a major challenge in the field includes non-invasive detection of small changes in fibrosis. Next slide. And with that, I will thank you for your attention and I look forward to the discussion and the further talks today and tomorrow. Thank you again.
Can I have my first slide? So it's my job to talk about the histologic aspects of assessment of fibrosis and how to assess reversal or progression of fibrosis, both of which are important and related topics. Next slide. This is my conflict of interest statement. I have a couple of uncompensated collaborative projects with HistoIndex and HiTide. Next slide. So this is the fibrosis staging system that we use in the NASH Clinical Research Network, and it's the one that is used by a number of clinical trials outside the network. There is an alternative fibrosis staging system, which is a little simpler and collapses those substages of stage 1 into a single stage. But what we're talking about today is changing stage from one biopsy to another. So the most basic situation is when a biopsy meets criteria for one stage in the first biopsy and meets criteria for a lower stage or a higher stage in the second biopsy. So next slide. This is the fibrosis pattern that you see in non-alcoholic steatohepatitis, or just steatohepatitis in general. It is quite complex, and you can see, so this is a Masson stain, so the blue stain is collagen, and you can see that there is perisinusoidal collagen wrapping around individual and small groups of hepatocytes. This collagen network connects in this particular photo between a portal area and a central vein, so technically bridging fibrosis. It's usually most prominent in zone 3, so it begins around that central vein and then radiates outward. And as in this photo, the bridging can be predominantly perisinusoidal. Next slide. So when I think about fibrosis improvement or worsening, I think about it in two situations. One is on the individual patient level, and one is kind of what we're talking about today is assessment across a cohort in a clinical trial. Next slide. So for one patient, it's easy enough to compare the slides side by side, and you can look to see whether it looks like there's a real difference in the fibrosis pattern. This gets around some of the, putting fibrosis into particular stages. So you can assess all of those features that we think are important in fibrosis side by side. Has anything changed in the same direction? So if the fibrosis looks worse, does the disease look worse? If the fibrosis looks better, does the rest of the disease look better? And then I include that sort of thing as a summary statement in my report. Next slide. So here is an example two patients that I photographed from our PIVENS study in the NASH network. Across the top, you can see before and after in a patient who improved their fibrosis from the first biopsy to the second. And what you can see is that there is less blue, and what blue that you see in the photo on the upper right is more consolidated. It's a little chunkier, it's shorter, it's not clearly as perisinusoidal as what we had before, but there's still fibrosis left. And I'm sure that these were actually different stages because that's how I selected them for photography. But, you know, just on a one photo level, it doesn't look stage-wise all that different. But other things are changing. There's less steatosis. There was almost certainly less ballooning in this patient and probably less inflammation. On the bottom photos, what you see is the reverse situation. So this was a patient who progressed between the two biopsies. And on the left, the bottom left, you can see that there's very little blue, maybe a thin little blue strand around the central vein in the center of that photo. And there's steatosis all around it. On the panel in the lower right, what you see is an extensive network of perisinusoidal fibrosis, lots of inflammation. There are some balloon cells in that photo, and there's more steatosis. So not only has the fibrosis progressed, but the rest of the disease has progressed as well. And I think we'll touch on this later in Dr. Bailing's talk. Next slide. So now for a cohort in a clinical study, you can still do that side-by-side comparison, but oftentimes there are lots of slides and that's not so easy. But you can still look at the data displayed in a grid. And this is the way that I like to look at clinical trials. You look at the general patterns, what patients shifted in one direction or another, how many didn't change or appear to change at all. Are there patterns in what you're seeing? Inconsistencies? Is there a dramatic shift at the high end or the low end of fibrosis? Next slide. So these are the changes in fibrosis displayed in these grids for two major NASH CRN pathology analyses. The one on the left was the one that we did to look at sort of natural history, included placebo arms and all of our database patients. And the one on the right was just treatment arm patients in PIVENS and FLIT. And so the stage at the first biopsy is along the vertical axis, and the stage at the follow-up is along the x-axis. And what you can see is that the red or orange color, those patients have apparently progressed. The green color, they've reversed. And the blue signals no change. So we have kind of a funny looking diagonal there because of the substages that we use in the NASH CRN. But I think there are a couple of things that you can take away from these grids. One is that there are a lot of patients who regress as well as progress in these cohorts. And some of the things that progress or don't progress or don't regress are patients around the high end. So if you look at the patients who started at stage 4, which is cirrhosis, none of them reversed past bridging. So none of them get down to those lower stages, but we have 6 patients on the left who reversed fibrosis from stage 3 to stage 0, no fibrosis, over the course of this natural history study. And the same thing happened in PIVENS. Another thing to take away from this is that we don't have much shift in stage 1 between the patterns that are pericentral, which are 1A and 1B, and the pattern that is only periportal, 1C. Basically, just a couple patients in each group flipped back and forth between these two stages, and that probably represents noise. Next slide. So these are our bar graphs that were also part of our natural history study. And what I want you to take away from this is, again, this relationship between fibrosis change and the change in the other things that are going on. So this is by diagnosis. And the diagnosis in fatty liver disease is based on a constellation of features. So in the upper left, we have disease diagnosis improved. So NASH going to NAFL, Borderline, we have an intermediate category going to NAFL, or NASH going to this borderline category. And what you can see is that in every pair of graphs, there are more patients with less fibrosis. But interestingly, in each pair, what you see is that there are a few more patients with cirrhosis. So despite the fact that patients have apparently improved their diagnosis, there does seem to be this subgroup of resistant patients who progresses. And similarly, at the other end, the larger bar graph over on the right, the NAFLD-NASH diagnosis worsened. You can see that this can be quite dramatic. So when you go from NAFL on the first biopsy to NASH on the second, almost all the patients who had zero fibrosis to begin with now have some degree of fibrosis. And at every one of these transitions, you can see that there are more of the higher stages represented. Next slide, please. This is the relationship between changes in disease activity and the stage change. And they're nearly linear relationship here, which was very interesting to us and completely unexpected to me. But what you can see is that patients who don't change their overall activity score, so change in NAS of 0 or plus or minus 1, don't change their stage much. It's still close to 0 change, but going either in the positive direction or in the negative direction, now you see more and more patients, and the whole average of the cohort either increases or decreases with changes in activity. So there's this relationship between disease activity and fibrosis that I think is very important and shouldn't be discarded. So there are other things that we can look at with fibrosis regression. And you know, when you ask, what does fibrosis regression really mean histologically? Are there clues that we can look at in the absence of side-by-side biopsy comparison? Should we flag it when a study entry biopsy shows evidence in fibrosis regression? And in conversation with my colleagues who are doing clinical trials, they are seeing biopsies right at the beginning that show these changes. Next slide. So again, just to remind you of potential fibrosis progression regression pathways, it's quite complex. It's really not linear in non-alcoholic fatty liver disease or in alcoholic fatty liver disease for that matter. You can track to bridging along multiple pathways, and one can imagine regression along multiple pathways, even if all we were doing was taking away fibrosis in the last place that it had been added. Next slide. Fibrosis regression is not a new topic. Ian Wanless published this seminal paper more than 20 years ago now, looked at cases of cirrhosis that were incomplete or complete for features of what he called the hepatic repair complex. And I'm not going to go through all of these features, but what I want to point out is the ones at the top of this table are probably the most important. The presence of perforated delicate septa, isolated thick collagen fibers, incomplete and thin spikes.
David, Naga here. I think there's a mismatch between what you're saying and what's the slide.
Oh, sorry.
Up.
Next slide. Sorry about that.
No problem.
I advanced my slide and didn't advance the one on the screen. All right, so let me begin again. So this is Ian's classic paper on fibrosis regression, and he looked at a whole bunch of features as part of this hepatic repair complex, including these changes in fibrosis that are actually fairly characteristic and easy to see. And I'm going to show you some examples of that. Next slide. So here are some photos from that original paper showing what he felt were features of fibrosis regression. And what you can see are very thin, very regular septa. And if you look at the panel on the left, there's a septa across the middle, sort of 2/3 of the way from the bottom, but then a really, really thin one very close to the bottom of that left-hand panel. And those kind of fibrotic septa are unusual in progressive disease. There's very little inflammation in them. The collagen is very well organized, and they are very thin. In the one in the middle, what you can see an example of an interrupted septa. So if you follow the very thin septa in the middle of that panel from top to bottom, about 3/4 of the way from the top, the fibrosis septa ends and then picks up a few cells later and then connects to the portal area at the bottom. So that's an interrupted septa. Next slide. So these are from 3 different patients. So I'm not trying to show you regression in one patient, but this is how he imagined things changed. You start off with something that is clearly cirrhosis, and then gradually you get thinning of the septa. They become less inflamed. You get those anti-fibrotic forces working to heal those fibrotic septa. And gradually they thin out, they become interrupted, and they disappear. But the underlying nodularity may actually stay around. Next slide. So this was applied a number of years later to a group of patients with chronic hepatitis B, and they evaluated patients after treatment as either looking at the fibrosis as either predominantly progressive, predominantly regressive, or indeterminate, meaning they couldn't tell. And the regressive ones were the ones that had the features that I just showed you. So if you go to the next slide, what you will see is some of their examples. And again, focus on that bottom predominantly regressive panel. So these were all patients who started with advanced fibrosis. And what you can see on the reticulin stain on the right is that the septa are very thin, and there's hardly any inflammation visible on the H&E on the left. In the indeterminate one, you can see a couple of these septa to the left and to the right of that middle right panel. But then in the middle of the panel, there's still a very active septa. It's very irregular. You can see that there's inflammation still in the middle of that biopsy. And so that one has some features of progression still as well as regression. Next slide. So you can see this in NASH. And here I have a couple of examples. One of the other clues that we use to say that there's fibrosis regression is that these are very difficult to stage sometimes. It's very difficult to put these changes into our standard definitions. And you can see in the example on the left that we have kind of a thin septa that sort of forms a tangential relationship with the central vein. So we aren't seeing that network of perisinusoidal fibrosis radiating outward. It's just this very thin septa. It doesn't seem to go anywhere. It doesn't connect anything. Probably a regression feature. On the right, we see kind of chunky collagen in the center. There may be some activity left because you can see some balloon cells there on towards the right of that picture. But then above the central vein, you see a short septa that starts and ends nowhere. It's just out there in the middle of the parenchyma, unable to connect to anything. And so again, that's probably evidence of regression, maybe kind of one of these indeterminate cases where you have features of both, but there's definitely regression going on. So next slide. So it is possible to detect features of fibrosis regression on a single slide. Sorry, I left out a word there. We look for these chunky collagen fibers, interrupted septa or bridges, or thin septations of mature collagen with little or no inflammation and no ductular reaction. And so in addition to staging in clinical trial situations, it may be worth flagging that biopsies have features of regression. And I think that would be a helpful thing when we're evaluating the data from these clinical trials. That and combining evidence of fibrosis regression with changes in the other features, because I don't think you're going to get fibrosis regression unless you change the underlying disease, which is what happens when you treat viral hepatitis in the best way. So thank you. That brings me to the end of my talk. If you go to the next slide, that's my final one. So thank you very much.
Thank you, Don. It was a great talk. Next slide, please. I want to thank the FDA for asking me to talk about NASH resolution. What does that mean for a hepatologist? Next slide, please. These are my disclosures. I served as a consultant to several pharma undertaking NASH clinical trials, as well as being part of many clinical trials, especially in NASH space. Next slide, please. So this is the outline of my talk. We'll touch on what is NASH, its relationship to fibrosis, its relationship to outcomes. How is NASH diagnosed? What is NASH resolution/regression? What is the significance of NASH resolution? Next slide, please. What started nearly 25 years ago, NAFLD was sort of deemed as a dichotomous. You have NAFL, which is benign. You have NASH that can progress to cirrhosis and liver cancer, liver failure. Sort of has evolved into subphenotypes. What you have on the left side is NAFL, which continues to be histologically benign from the liver standpoint, and far right is NASH cirrhosis. In the middle is NASH with varying degrees of fibrosis. I want to draw your attention to a couple of things. One is underlined. The liver disease as the primary morbidity and mortality occurs in NASH with F2 or higher fibrosis. Whereas cardiovascular disease and cardiovascular events feature quite heavily for NAFL, NASH with early fibrosis or no fibrosis, as well as NASH with advanced fibrosis as well. So if you're trying to improve the outcomes of patients with NAFLD, not just the liver disease, but also cardiovascular entities become important. Next slide, please. As I was preparing for this talk, I was just looking back at the literature about the NASH natural history, some of the earlier data. What I have here are three, I would say, fundamental articles published between mid-80s to mid-90s. The first one is from Elizabeth Powell and colleagues from Brisbane. The next is Anna Medill, Zach Goodman, and Kamal Ishaq from AFIP. The third one is Bruce Bacon and colleagues from St. Louis University. These papers have made clear that the NASH is progressive, can lead to cirrhosis, liver failure, but interestingly, none of these papers used ballooning to define NASH. Really, the Bruce Bacon's paper in 1994 in gastroenterology defines non-alcoholic steatohepatitis as steatosis with inflammation, with or without hepatocyte necrosis, Mallory's, hyaline, and fibrosis, no ballooning. Next slide, please. Sort of the ballooning and sort of features in what I call really a seminal paper published in 1999 gastroenterology from Cleveland Clinic. Art McCullough was the senior author. Zubair Younasi at the time was at Cleveland Clinic. I think this sort of fundamentally helped us to how we design trials over the course of the next 25 years or so. What they've done in this trial, in this particular study, this was a retrospective study, 135 or so patients with outcome data up to 18 years or so. They have retrospectively classified liver histology into 4 subgroups. Type 1 is fatty liver alone. Type 2 is fatty liver fat accumulation with inflammation, lobular inflammation. Type 3 is fat accumulation with balloon degeneration. Type 4 is fat accumulation, balloon degeneration. Either you have Mallory's, hyaline, or fibrosis. This is truly an important paper. If you look at the table 4 on the right side, development of cirrhosis is almost exclusive to type 3 and type 4. And so our liver-related deaths are more enriched in type 3 and type 4. This sort of formed the basis for some of the work subsequently was done by the NASH CRN. Next slide, please. This paper, Dr. Kleiner was the first author, has been cited over 5,000 times, is sort of remains the basis how liver histology, both in clinical trials as well as in clinical practice, is scored in the United States. This paper is the first time that I know mentions of definite borderline or no NASH terminology. The figure illustrates a couple of things. One is on the x-axis is different histological features. On y-axis, you see the proportion of patients with steatohepatitis. What I draw your attention to is about 10% of the patients with a diagnosis of steatohepatitis do not have ballooning. That's one. The second point is most patients, at least in this particular study with steatohepatitis, have fibrosis. I think under 10% are without fibrosis. And then the last point I'd like to make here is in this particular paper, NAS of 5 or higher correlated with the diagnosis of NASH, and the biopsies showing NAS of 3, less than 3 was not NASH. Next slide, please. So this is another sort of a landmark event, 2010, 2009, AASLD had an endpoints workshop that many in the panel elsewhere are part of this. At the time, though, if you're doing clinical trials in NASH, what is an endpoint? And we sort of thought, if somebody has NASH going into the trial, not having NASH is sort of a good outcome. You no longer have NASH. And there was a debate about how do we define definite NASH and what is sort of resolution of NASH, sort of the first discussed at this meeting, and this paper was published in Hepatology, I think in 2011. Here you see ballooning, hepatocyte ballooning is a must feature for definite steatohepatitis, and also this mentions about what is not hepatosteatohepatitis with or without steatosis. In the footnote, I quote from the paper, subjects with steatohepatitis are more likely to develop cirrhosis and experience liver-related mortality than those with fatty liver alone or steatosis and inflammation. Therefore, reversal of steatohepatitis with no worsening of fibrosis or actually improvement in fibrosis is a reasonable surrogate for prevention of cirrhosis and liver-related death, unquote. Next slide, please. As I already touched on, NASH and fibrosis tightly coexist, except for patients who have advanced fibrosis, i.e. burnt out NASH cirrhosis. Otherwise, you always see active disease. And you can sometimes have active disease with trivial fibrosis, even that's not necessarily the case. There are 3 papers that I listed here, Cleveland Clinic paper, a seminal paper from Paul Angulo in gastroenterology, as well as the JAMA Network open paper Dr. Kleiner just elaborated, all make this point, NASH and fibrosis tightly coexist. Next slide, please. This was already touched on by Dr. Kleiner. Once again, I sort of want to highlight that in this particular paper from NASH CRN, where natural history of liver histology was looked at in paired biopsies from the NASH CRN, 3 points. One is at baseline, 25% of patients deemed to have NAFL had some fibrosis. The second is even NAFL patients had, and about 45% had an increase in fibrosis by a stage on second biopsy. And then third point is whether you have borderline or definite NASH, the risk of progression to cirrhosis is similar. Next slide, please. This is, once again, the same data set Dr. Kleiner touched on, showing that histological progression of fibrosis is highly related to disease activity progression. Here, if the disease activity progressed, the odds ratio for fibrosis progression is 7.2-fold higher. with somewhat broad 95% confidence intervals. Next slide, please. So what I've shown so far is that the steatohepatitis exists with fibrosis, and if the steatohepatitis is worsened, there is more likelihood of fibrosis worsening. And this particular paper from Paul Angulo, Dr. Kleiner was the second author on it, showing that baseline steatohepatitis is significantly associated with long-term outcomes. And there is discussion in the field that when you compare fibrosis versus steatohepatitis, generally fibrosis is thought to be in the hierarchy of things far more important. That doesn't diminish the importance of steatohepatitis. Here, if you have definite steatohepatitis or borderline steatohepatitis, you're far more likely to have poor outcomes. This is an unadjusted univariate analysis. In the bottom table, or I have an editorial listed here from Professor Vlad Ratsu, objecting to, when you're looking at the importance of steatohepatitis, he objects to controlling for fibrosis because they are so coexistent, which I think is a really valid point. Next slide, please. How about improvement in steatohepatitis is strongly associated with improvement in fibrosis? This is a paper, once again, from the NASH CRN. Dr. Brunt was the first author. Dr. Kleiner was a key member of this particular team. Fibrosis improvement in 2 clinical trials conducted by the NASH CRN, both PIVENS and FLINT, was most significantly associated with the resolution of NASH. Not only resolution of NASH, even improvement in NAS by 2 points or higher correlated with fibrosis improvement. You see the odds ratios there. Next slide, please. In terms of relationship between resolution of NASH and the incidence of cirrhosis and clinical outcomes, although biologically quite plausible, at this time there aren't robust published data to confirm that resolution of steatohepatitis lowers the risk of development of cirrhosis or clinical outcomes. Data from Regenerate trial with obeticholic acid, as well as Maestro NASH phase 3/extension trial with resmetirom, hopefully will provide that confirmatory evidence to the significance of NASH resolution and improving the outcomes of patients with NASH. Next slide, please. To summarize, NASH, a constellation of steatosis, inflammation, and hepatocyte injury, highly coexistent with fibrosis. It is associated with fibrosis progression. It is associated with development of cirrhosis and liver outcomes. NASH resolution is highly predictive of fibrosis improvement. However, in terms of development of cirrhosis and outcomes, we are still waiting for confirmatory data. Next slide, please. As part of this exercise, I curb-sided about a dozen of my colleagues, hepatologists and one endocrinologist. Summary is that they uniformly believe that the steatohepatitis is a key driver of fibrosis in NASH. Resolution of steatohepatitis is a highly meaningful endpoint in a patient's journey. Several expressed frustration practically documenting resolution of NASH in clinical trials, primarily in documenting identifying balloon hepatocytes. Few recommended reverting back to NAFLD activity score-based approach to document NASH resolution. Last slide, please. Hepatologists view NASH resolution as reverting back to NAFL or non-NAFLD, and thus significantly lower risk for fibrosis progression and cirrhosis development in patients with non-cirrhotic NASH. I want to thank you for your attention and like to call upon Dr. Belling for her talk.
Hello, I'm Cindy Bailing. I'm a liver pathologist, and I have been asked to speak today about the pathologist perspective of NASH resolution. Next slide. Can you advance, please? Sorry, I think we missed. I think we missed my conflicts of interest. I am an employee of Pacific Rim Pathology Lab. My lab has service contracts for technical and professional services with ICON, LabCorp, Perspectum, MedPace, and Sharp Healthcare. And I've provided educational lectures to Novo Nordisk, Alimentiv, and Pfizer. And I currently consult for Hologic Pathology Institute. And I am currently scoring or have scored several NASH trials. Next. Okay, that's good. Actually, back up one, sorry. Dr. Siegel went over the liver biomarkers at length, but I'd like to frame his remarks from a pathology point of view. So liver biopsy as a biomarker enables measurement of all the key features that are required of a biomarker. Tissue evaluation allows direct observation of all the cell types and matrix interactions in the target organ, provides the diagnosis or confirmation of disease and exclusion of other diseases. It can relate severity and identify features associated with outcomes. In addition, biopsies can demonstrate potential safety signals in the form of known histologic changes and allows assessment of disease remission or regression. Correlation of phenotype with prognostic or other features may eventually also allow biopsies to identify best treatments for individual patients out of a spectrum of effective therapeutics. Next. As a reminder, the features of NASH, which are measured in biopsies and encompassed in current scoring systems, are relevant to the biology of the disease. Steatosis correlates with insulin resistance and impaired glucose metabolism. Inflammation stimulates fibrosis. Ballooning degeneration denotes hepatocyte injury and correlates with worse disease. And fibrosis itself correlates with liver-related outcomes and mortality. So in one biopsy, one can view all of these correlates together. Next. The classic morphologic pattern of NASH featuring steatosis, hepatocyte injury, and inflammation with or without fibrosis suggests different therapeutic targets. The appearance of NASH resolution might vary depending on the targeted feature. Shown here are 3 examples of typical NASH, each demonstrating steatosis, lobular inflammation, and cell injury in the form of hepatocyte ballooning. Before I show you examples of what NASH resolution looks like, I want to clarify that when I use the term resolution here for this talk, I'm not implying a permanent ending of the disease. And I also want to convey that a biopsy might show significant therapeutic effect, but yet not meet current definitions of resolution as we have stipulated them. Next. In its most complete form to a pathologist, NASH is resolved when all the features of disease are no longer present. And you can see an example of a resolved NASH biopsy in the slide here. Since therapies act on different components of the disease, however, in a post-treatment biopsy, some histologic findings may still be present to some degree. And I think you can see that in this biopsy, there's a minimal amount of steatosis that's residual after treatment. Next. Using NAS scoring, we're going to go through in the next series of slides, we're going to go through each component of the NAS and what it looks like when it improves or disappears. So using the NAS scoring definitions, steatosis improvement is a reduction in the percent of hepatocytes containing fat droplets. So this is an example of a paired biopsy where the left is the screening biopsy, which was scored as an S3 or severe steatosis. And post-treatment on the right, you can see the steatosis has markedly decreased and would now be scored as an S1. It is possible also to reduce steatosis, but not to a degree that changes the steatosis grade. That is, there might be intra-grade improvement in steatosis. This is true for actually all of the NAS components. In a direct comparison, such as we have here, you might see an improvement, but yet the score feature remains the same category. Also notice that in addition to the large droplet fat, we can see some small droplet fat in the biopsy on the right post-treatment, which raises the possibility that the fat droplets are actually getting smaller but have not yet disappeared. Next. While a simple reduction in steatosis is a relatively straightforward assessment, reduction in the size of the fat droplets, the presence or location or shift of the steatosis to a predominantly small droplet fat pattern, and in some cases, the presence of microvesicular fat are changes that are a little more difficult for pathologists to characterize. These three biopsies illustrate some of these alterations in treated patients. So on the left, we see a more diffuse distribution of steatosis rather than the sort of typical NASH appearance of a central lobular or zone 3 emphasis for the steatosis location. In the middle, we can see that there's a mixture of large and small droplet fat in the hepatocytes. And on the right, it's a little hard to tell from this photograph, but there's microvesicular steatosis in the background as well. So these are three changes that we see that do not, we see these in treated biopsies. They do not necessarily translate perfectly to the current scoring criteria. For these types of changes, complementary image analysis methods might be useful, and we're going to hear more about those tomorrow. The histogram that I show is just an example of a fat droplet size distribution analysis and demonstrates how image analysis might be more sensitive for detecting these smaller changes in steatosis. Next. To the right is an example of a treated patient with near complete disappearance of large droplet fat after treatment. The photos on the left show some large lipogranulomas, which have not previously correlated with anything in steatotic liver disease. But it serves to illustrate the point that there are features that we see in NASH patients and in clinical trials, which may be present but are not incorporated in current scoring systems. So the current scoring with steatosis, ballooning, and inflammation are really only three of many pathologic findings that are present in biopsies with steatohepatitis. Next. So we'll turn to lobular inflammation. The current NASH resolution criteria require an NAS lobular score of 0 or 1 for lobular activity. This is enumerated as fewer than 2 foci per 20x field in the microscope. And generally, pathologists in our discussions over definitions and how to characterize a score of 0 for lobular, most pathologists agree that if you only have 1 or 2 foci of lobular inflammation in a centimeter of biopsy, that warrants a score of 0 for lobular inflammation. So on the left, we see these are again paired biopsies from a patient who's been treated. So on the left, there's about 4 large foci of inflammation. And I picked this stain because you can kind of see they have a little more bluish tinge. They don't always look like that, but this was kind of nice for illustration. So there's at least 4 foci in this field on the left. And in the paired biopsy from that patient on the right, after they've been treated, we don't see any foci of inflammation. Next. Looking at ballooning, in the best circumstances, the balloons disappear after treatment. So on the left, we can see classic ballooning degeneration, which is what most people think of when they think of ballooning ballooned hepatocytes. And so that's pretty obvious. So in this paired biopsy, you can see some balloons right in the middle of the screening biopsy on the left. And then the post-treatment biopsy on the right, there are no remaining ballooned hepatocytes. So that's kind of an ideal situation where it becomes very clear that the ballooning has resolved. In this patient, the steatosis has also improved, although that's a little bit more of a subtle finding in that particular patient. Next. This is a more subtle example of probable balloon resolution. You can see in the biopsy on the left, which is the screening biopsy, there's a cluster of cells around the central vein that are abnormal, but don't, they're balloons, but they're not quite as prominent or obvious as the ones we saw in the previous slide. However, in the post-treatment biopsy on the right, there are no balloon hepatocytes. And one of the things that pathologists do is we very carefully look at where we look at the whole biopsy, of course, but we also emphasize looking at where we expect the balloons to be. So in a biopsy like this one on the right, we would very carefully examine all of the central vein regions as a confirmation that really we aren't seeing balloons in this patient. One of the things you can also note in this particular paired biopsy is that there's a slight shift in the fat droplet size. So there's still a little bit of fat left in the treated patient on the right. The fat droplets look smaller than the ones on the left. Next. So image analysis studies, and particularly the paper last year from Beth Brunt and others, really demonstrate that there's a wide range and a complexity of appearances of balloon hepatocytes. Balloons might be small. They may not have all the features that we typically would like to see. And these pictures sort of illustrate that point. So ballooning degeneration is present in both of these biopsies, but the biopsy on the right in the lower square shows some cells that we would consider non-classic balloon hepatocytes. So they're likely balloons. They don't have all the features of a classic balloon. This brings up the issue that the life cycle of a ballooned hepatocyte related to the morphology is not very well understood. We don't really know how long they persist in tissue, and we don't really understand completely how the features of a ballooned hepatocyte recede or what they look like when they're in the process of resolving or say early resolved. And the NASH CRN has addressed this because we recognize this issue in our treated patients and have published an expanded balloon score, which we hope will capture some of these what we call non-classic balloons. That is, cells that have the features of balloons in the right context, but perhaps don't have all of the classic features. And we see these in the clinical trial setting. And so I think it's important that we attempt to resolve the balloons as well as resolve the NASH. Next. These are just a few more additional examples of changes in ballooning that have been observed in the treatment setting. So on the left are a few more non-classic balloons. They're smaller, less distinct than a classic balloon. And then another finding that we occasionally see are the sort of persistence of balloons in the tissue. So the fat has disappeared, the inflammation is minimal, but yet we still see these ballooned hepatocytes. Again, this may relate to the temporal nature of how long it takes for a balloon to disappear once a patient has been treated, but we really don't completely understand the pathophysiology of ballooning resolution. Next. It's important to note that while the NAS score reflects disease activity, the SUM score does not entirely reflect the pattern of disease. Shown here are two cases with an NAS score of 5. In the first panel, which is A on the left, there's very mild steatosis, but notable ballooning degeneration and moderate lobular activity. In the second case on the right, there's severe steatosis, but only mild lobular activity and very few balloons. These two cases may respond differently to a drug. Next slide. We also know that portal tract changes correlate with NASH severity, but they are not currently included in commonly used scoring systems. Incorporating portal tract inflammation into histology assessments might eventually add useful information. Next. Despite the fairly straightforward appearance of successfully treated NASH, there are many things that we don't know about NASH resolution. Is any particular histologic feature more important? Does it matter how we change the disease? Should there be sequential histologic targets? How fast do the histologic improvements happen? And do the histologic features regress at different rates? This tempo of pathologic improvement is important to consider as we try to shorten the time to demonstrate improvement in clinical trials. Some drugs might erase pathologic features faster than others. And we also see evidence for reprogression of disease in previously treated patients, but this also has not been adequately studied. And next. Last slide. To answer the question, what does resolution look like to a pathologist? We can say a partial or near complete reversal or disappearance of the pathologic findings. There are, however, a number of unanswered questions regarding NASH resolution, which can be studied using biopsy and which will help us understand the disease and treatment effects. These questions are likely important in understanding therapeutic effect on outcome and can be understood using biopsy analysis and histologic endpoints. Thank you for your time. I look forward to the panel discussion.
Next slide, please. The topic of my talk is the tension between surrogates and clinical benefit. Next slide, please. I'm chief of the translational hepatology section. I have no disclosures. This talk is going to be conceptual, and after discussion with a number of people, is going to stay away from the weeds of a lot of the NASH data and instead sort of stick to a higher level overview of that tension that we already alluded to. Next slide, please. This is taken from the FDA. A surrogate, and you've heard this already, a surrogate endpoint is a clinical trial endpoint used as a substitute for a direct measure of how a patient feels, functions, or survives. The surrogate endpoint does not measure the clinical benefit of primary interest in and of itself, but rather is expected to predict that clinical benefit. In general, the predictive nature of a surrogate endpoint is determined through the evaluation of epidemiologic, therapeutic, pathophysiologic, or other scientific evidence. Next slide, please. So I want to ask a different question. I want to ask, what is it that we expect of our surrogates? In very simple terms, that's very elegant language, but what we really want is for the surrogate to predict an endpoint. And more than that, and I think this is equally important, an alteration in the surrogate should result in an alteration in the endpoint. And I'm going to discuss this in some detail. Next slide, please. There are different kinds of surrogates. The candidate surrogate endpoints, reasonably likely surrogate endpoints, and validated surrogate endpoints. And we'll quickly go through two of those and then discuss the third in more detail. Next slide, please. So before we do that, I'd like to discuss some theory. When we talk about a disease like MASH, there are multiple initiating events which lead to the disease initiation. In hepatitis C, it's simple. It's the virus. I think it's a little bit more complex here. Regardless, once the disease initiates, that should lead to an ideal surrogate endpoint, something that is in the direct line of development of a clinical outcome. And the clinical outcome is where the benefit potential lies. Next slide, please. If this is the case, where disease initiation leads to surrogate endpoint, which is directly related to clinical outcome mechanistically, the R squared, or the relative effect, is 1. And there's been an extensive literature looking at the statistics of this. And there are different ways to measure this, which I'm not going to discuss, other than to say this is the ideal, a 1. Next slide, please. However, life's a bit messy. Not everything is that clean. And surrogate endpoints can be associated with the clinical outcome before the initiating events, during the initiating events, or actually after the disease initiation. And if someone with lung cancer or cancer associated with poor socioeconomic status wins the lottery, it's not going to cure them. Next slide, please. And here the R squared or relative effect would range from 0 to 1. 0 when there's no real relationship in terms of cause and effect, and 1 where there is that relationship we already discussed. Next slide, please. Candidate surrogate endpoints are being evaluated for the ability to predict clinical benefit, and I'm not going to discuss them further because that's not what we're discussing. Next slide, please. Validated surrogate endpoints are supported by clear mechanistic rationale and clinical data providing strong evidence that an effect on the surrogate predicts a specific clinical benefit, and it's typically directly related to the clinical outcome. And I would argue that if an endpoint like this is available, conducting a clinical endpoint study may be unethical. However, we're not going to discuss this much further today. I'm not going to discuss this much further either. Next slide, please. I would like to say that validated endpoints are often ideal, often even idealistic, and the enemy of good is better. By striving for something that is perfectly validated, we may not get or even start to go where we'd like to be. Next slide, please. However, where we're going to spend some time thinking and discussing is a reasonably likely surrogate endpoint. And here are the FDA definitions. Reasonably likely surrogate endpoints are supported by strong mechanistic or epidemiologic rationale, but the clinical data available is not sufficient to make it validated. And a reasonably likely surrogate endpoint can be used to support the FDA's accelerated approval program. And it's intended to provide patients with serious diseases more rapid access to promising therapies. So this is an answer to that tension. This is an attempt to resolve that tension. Next slide, please. There's a cost-benefit balance. If we use a reasonably likely surrogate, it may not actually predict. It may not actually be reliable at all. And we may miss delayed safety signals because our surrogate is met and the safety signals occur later. And effective therapies may be delayed because we focus on using drugs that may not work. Other clinical trials are stopped. And instead we focus on this. Next slide, please. However, using reasonably likely surrogates results in efficient, smaller studies with decreased time needed for drug approval and gets therapies to patients faster. You see the tension continues between the cost-benefit balance. Next slide, please. We have to ask why surrogates fail. There are multiple reasons. There's a lot of literature regarding this. Association does not equal causation. Because it happens before the event does not mean it caused the event. Temporality does not equal causality. And intervention to improve the surrogate, however good the surrogate may look, may not afford clinical benefit. And we really have to understand the driver. The body shop is not the solution for bad driving. Next slide, please. And it happens. And if we think failure of surrogates doesn't happen, it really does. I want to look at 3 fields. I want to look at cardiology, endocrine, and oncology. Next slide, please. Cardiology has lots of good. Hypertension predicts stroke. LDL predicts cardiovascular events. These are well-accepted surrogates which are mechanistically linked to outcomes and have been shown over and over to predict outcomes. Next slide, please. However, increasing LDL is not as clear. And in fact, there may be some evidence that as LDL is increased further, it may be harmful. Suppression of PVCs by class 1 antiarrhythmic agents post-MI increases mortality. It's a confounder for left ventricular function. Postmenopausal hormone therapy improves lipid profile, but does not change cardiovascular events. And these are all examples of surrogates which in different settings may predict outcome or may not predict outcome or are strongly associated. But in the trials and validation studies failed. Next slide, please. I want to focus on just one example in endocrine because it has been so well studied and so well characterized. And in the paper by Cohen, he really dissects the whole story. Rosiglitazone was approved by the FDA in 1999 and EMA in 2000 based on short-term phase 1 to 3 clinical trials. And the surrogate endpoints that were improved were blood glucose and glycosylated hemoglobin. There are still medical students today who are being taught that improving blood glucose decreases risk of complications. And that's true. But meta-analysis of randomized controlled trials 10 years later and in the record trial of over 4,000 patients with a primary outcome of cardiovascular hospitalization or cardiovascular death showed that the addition of rosiglitazone to standard drug therapy did not improve cardiovascular risk, was associated with increased heart failure hospitalization and a potential increase in myocardial infarction, and the drug was withdrawn in 2010. Next slide. In oncology, I just want to mention one systematic review, which was published in JAMA Internal Medicine in 2015. The conclusion stated, most trial-level validation studies of surrogate endpoints in oncology find low correlations with survival. All validation studies use only a subset of available trials. The evidence supporting the use of surrogate endpoints in oncology is limited. Next slide, please. And this is some of the data where you could see that the low evidence was mostly focused around metastatic disease, which is what matters most in oncology. Next slide, please. In an accompanying editorial, faster drug approvals are not always better and can be worse. Dr. Redberg pointed out that in the effort to get new drugs in the market more quickly, many agents are approved on the basis of surrogate endpoints, such as progression-free survival. Unfortunately, these endpoints do not necessarily translate into mortality benefits, but they do cause toxic effects without giving extra time. And she went further. Even when the US Food and Drug Administration takes the dramatic and rare step of withdrawing approval, as it should, when a drug approved based on surrogate markers later turns out not to offer mortality benefit, as in bevacizumab, for metastatic breast cancer, women will continue to be treated with this harmful drug, although we know it will not extend life. In our rush to find new effective treatments, we should not harm our patients with ineffective toxic ones. Next slide, please. And in case we think it doesn't happen, that this was now, that this was all 10 years ago, 20 years ago, McKenna was withdrawn this year, approved in 2011 based on a surrogate. Next slide, please. And in two really worth reading manuscripts by Chiani et al., a surrogate primary outcome overestimates health benefits of interventions by 40%, with an adjusted ratio of odds ratios of 1.46 compared to trials using a primary patient participant relevant final outcome. So we went through three fields, starting with specific examples, and now we're speaking very broadly about the overall concept of surrogates and studies related to surrogates. Next slide, please. So we have to separate confounder versus causality, the PVC and left ventricular function story. To be fair to surrogates, perhaps the trials were not long enough. Perhaps the dose was incorrect. Perhaps there was too small a population. Perhaps the population is incorrect. But the problem is it gives false hope, and it costs so much money, so much time, so much effort, so much emotional effort, and it delays real therapies. Next slide, please. Some history that I found really interesting is the Bradford-Hill criteria. Bradford and Hill were statisticians, and in the early years, there was argument about whether tobacco really was causal of lung cancer and whether it was an adequate surrogate, whether pack years were adequate surrogates. And they came up with criteria which they applied to the concept of whether or not a surrogate would be valid. They spoke about the temporal relationship, that the cause should always come before the effect, the strength of association, the dose-response relationship, and we saw that with amyloid today, consistency of the relationship, biological plausibility, consideration of alternatives, experimental verification, specificity, that there's a specific cause for a specific effect. And the problem with specificity is that this is, for me, the one criteria that is not really always applicable. For example, I think that there are many things that go into causing an elevated LDL. So this may not always apply. And lastly, they asked for coherence that's compatible with existing knowledge. Next slide, please. So let's go a little bit closer to the liver. And actually, we spoke a lot today about resolution of fibrosis. To my knowledge, the first real demonstration of cirrhosis resolving was this case report from Darrell, Jay Hufnagel, and David Kleiner, where a patient with delta hepatitis was treated for 12 years with interferon and cleared his hepatitis and his cirrhosis. Most people focus on the 12 years of interferon. However, I think this case report is very instructive for other reasons. The NITs, or non-invasive tests, did not improve initially. Look at the ALT. It essentially was elevated for 6 years. But the liver biopsy, the inflammation on the liver biopsy started to decrease almost immediately. And more than that, the virus is what was really driving the event, and that went away. Next slide, please. So here the driver is hepatitis D. And although not shown on this figure, the viral levels and the changes in the virus are what are really important here. The NITs would have been misleading, and this is looking at it in a different way, a different reason to be careful. Next slide, please. So what about steatotic liver disease? My belief, as has been alluded today by others, is that metabolic dysfunction-associated steatohepatitis resolution will lead to reversal of fibrosis and increase in clinical benefit. Whether that's true or not is kind of irrelevant, but I think that really is the case. But for the sake of discussion, let's just conceptually think about this. If that's true, how should that be measured? Next slide, please. So X+ associations, the very faint, hard to see because we don't really understand it, leads to MASH, which leads to cirrhosis, which leads to death. We can measure this by imaging, blood tests, and histology. Just as an aside, it's easy to say histology is difficult to do and not accurate. We could say it's not practical, but I would point out how wonderful the separation of curves are for single stages in fibrosis are. Looking at patients with MASH, how you can go from F0 to F4 and how the curves separate so beautifully clinically. If anyone thinks that fibrosis, a single stage fibrosis, is not separable by histology, those curves prove otherwise. So there are lots of ways to measure this and to think about this. In the absence of a clear driver like hepatitis C or hepatitis D, we can measure these things. We have to ask ourselves, what's a technical improvement versus clinical improvement? And this is something that David Kleiner always says. So if you have a certain change, as was alluded to earlier, 1 kilopascal on MRI, is that a technical improvement or is that a real clinical benefit? And this is an important thing to consider. Next slide, please. So we have to ask which are reflective of the line. What drives the progression across that line, going from initiation to death? And we have to ask ourselves, how are we going to compromise in the absence of a clear driver, like a viral load? And how are we going to think about coming up with reasonable surrogates? Next slide, please. There's another problem. How do we separate the driver from unavoidable collateral damage? There are multiple diseases. The four drivers, the four horsemen, I think of this as the four horsemen of liver disease, is that there's an underlying disease driver, there's inflammation, fibrosis, and portal hypertension. And these can all mislead us. For example, ferritin is released with inflammation. And there were studies looking at phlebotomy as a treatment for NASH. Well, phlebotomy removes a lot of calories, that's true, but it doesn't really stop the progression of NASH. And furthermore, the last three are tightly associated with the driver, but are confounding elements in the sense that they may also be causal of death, but not drivers of the underlying disease. That's not to say it's not important. For example, giving carvedilol to someone with clinically significant portal hypertension and delaying decompensation is very important, but it doesn't treat the underlying problem. You can drain the swamp, or you can shoot alligators. And I would prefer to drain the swamp. Next slide, please. What about on therapy? What about non-invasive tests once patients have started therapy? If ALT or AST are not the driver and therapy decreases it, non-invasive tests validated off therapy will not be valid and difficult to follow, because we saw that some patients still progress to cirrhosis on therapy, and that might be missed. Furthermore, as in the hepatitis D patient, the underlying driver may be improving, but ALT or AST may lag. It would look like there's no effect, and this is especially true for fibrosis. Next, if the surrogate truly reflects underlying mechanism, it would remain accurate. And this is what we should strive for. And again, even if non-invasive tests are true surrogates, they may not reflect a safety signal, especially if delayed. Next slide, please. Final thoughts, and these are mine alone. Reasonable surrogate endpoints allow faster drug development. And we can't stop that. We have to allow that. That makes sense. Trust is good, but validation is better. Proof of clinical benefit should still be required. The cost of being wrong is too high. Patients treated without benefit and continued disease progression cannot be acceptable. We have to advance the field, but we have to have reasonable checks and balances. It would be terrible to rush up a ladder and only find out you're against the wrong wall. Next slide. With that, I'll end. Thank you very much.
Thank you, Theo. That was a wonderful talk. My co-moderator, Dr. Johnson, and I greatly appreciate all the speakers, and also thank the audience for your interest. We will take a 15-minute break, and we will be back here at 12:35. Thank you so much. Welcome, everyone. My co-moderator, Dr. Laura Lee Johnson, and I invite you to panel discussion of Day 1, Session 2. As a reminder, the goal of the workshop is to assist FDA in identifying current knowledge gaps for using non-invasive tests as diagnostic biomarkers and reasonably likely surrogates, and provide a framework for the additional data needed to fill these gaps. The primary focus of the workshop is non-cirrhotic NASH/MASH population with advanced liver fibrosis. We're especially interested to hear from you about knowledge gaps in the current endpoints for NASH phase 3 clinical trials and how these knowledge gaps related to the NITs, and these biomarkers may be used as prognostic, enrichment, diagnostic, and response biomarkers. We plan to allow each panelist who has not given a talk about 3 minutes for introduction and presenting their initial thoughts, perspective, and response. And this should permit us about 30 to 40 minutes for Q&A panel discussion. We remind the panel participants to be professional and respectful. We're interested in an open scientific discussion on data exchange and data existing and needed to support the focus of the workshop. During Q&A, please limit your responses to 1 minute, and if you're interested, please raise your hand. The moderators will do their best to ensure that the conversation remains focused on the workshop and workshop's primary focus. With that introduction, I'd like to call upon Dr. Mary Rinella to give introductory comments. Dr. Rinella is professor of medicine in the Division of Gastroenterology, Hepatology at the University of Chicago. She's a key opinion leader in the NASH field, and she is the first author on the practice guidance from the American Association of Study for Liver Disease that was published in 2023. Maru?
Thank you, Naga, and thanks to the FDA for the invitation to participate in this extremely important panel and workshop. So as we heard from the speakers today who highlighted the importance of histology in how we assess this disease, I have a couple of thoughts. With respect to steatohepatitis, I think it's fairly clear that the low intra-observer variability in key histologic features, namely ballooning primarily, but also lobular inflammation, are part of what is driving the difficulty with defining the NASH resolution endpoint. And I do think, though, that if we were to retain liver histology as an endpoint to assess disease efficacy, then maybe we need to revisit how we define NASH or MASH resolution, bearing in mind this needs to be applicable and replicable across pathologists in different settings, or set a threshold to define NASH or MASH improvement, and perhaps AI can help us with this. With respect to fibrosis, there is a huge range of fibrosis burden, particularly at more advanced stages of disease, and that's going to be quite broad. And thus, this poses a challenge to show one-stage improvement in fibrosis, not to mention the fact that we know that from viral hepatitis, when the insult is completely quelled by treatment intervention, fibrosis improvement takes time and sometimes years. So there are clear examples of where substantial improvement in reduction or in fibrosis can occur, for example, in stage 3 or stage 4, where there's a broad range of fibrosis burden. And this can exceed that of moving from, say, F3 to F2 or F2 to F1, yet fail to drop a stage of disease, just as illustrated by Dr. Bailing earlier. So did this person benefit less from treatment than the other one who crossed the disease stage threshold but had less substantial fibrosis improvement? And I think this is a major issue for us. Histopathology obviously plays a key role in the diagnosis and treatment of liver disease, but on a broader scale, it's limited by risk, cost, et cetera. And so given the limitations of histology is the only accepted measurement of improvement in the current conditional approval paradigm, it really begs the question of why we continue to commit to histologic endpoints, and more importantly, how can we expect to have accurate biomarkers when the reference standard has so many limitations?
Thank you, Maru. I'd like to call upon Dr. Scott Friedman, who does not really need introduction. He's professor of medicine, the Dean of Therapeutic Discovery at Mount Sinai School of Medicine. He's the division head for hepatology. And he's the father figure in the biology of fibrosis, and his lab has discovered stellate cells as the source for fibrogenesis. Scott, please.
Naga, thank you, and thanks for the generous introduction. I was really delighted at the speakers who preceded us and touching on many of the key points. And I would like to look a little bit from a more scientific perspective and provide a little bit of comparison or reality check. First of all, we've been very spoiled in hepatology by starting to assess fibrosis and the improvement in the context of curative or suppressive therapies, hepatitis C, B, and Delta, which gives us set a very high bar for what we should expect in chronic inflammatory diseases. And I think one should, instead of looking to those diseases as examples, we should think about chronic inflammatory diseases in other organs. For example, inflammatory bowel disease, where initial therapies were partially effective, mostly blunts like corticosteroids. And increasingly, we've achieved a nuanced understanding of IBD to recognize which features will connote improvement or responsiveness and which will not. And that really leads me to the concept that in some ways, histology is really a rather blunt instrument. No one can question the primary role it's had in defining the disease and assessing progression or regression, but it really leaves a lot of granularity out of the picture. And one of the things we've learned from particularly single cell methodologies, is that diseases and cell types are extraordinarily heterogeneous. And I think we need to think about that heterogeneity in the context of NASH, meaning, is it one disease, or are there subtypes of NASH that offer different drivers and different therapeutic options, much like IBD is evolving? So, for example, we know there's heterogeneity in the cell types, in localization of fibrosis, in the biochemistry of fibrosis, and of course, in the nature of fibrosis and its regression based on the specific etiology we're exploring. And they are probably going to be different. We're still in a very primitive stage in understanding the biology or biochemistry of matrix degradation. We don't know where the proteases are, what cells express them, how they are regulated, how important they are, and whether they are equal no matter how fibrosis regresses. And here I would refer to the discussions already and to follow around AI, artificial intelligence, because I think in the end, we are going to need to incorporate complex data sets, both from genetics, from histology, from clinical variables, blood tests, in a way that I don't think we can just integrate through our own efforts, but rather have the data filtered through an AI-based approach. Finally, in terms of histology, as we'll hear later, I think along the theme of histology being a blunt instrument, there's no question in my mind that the more we understand the nuances of fibrosis, the more we're going to have to rely on digital methods and AI-based methods to quantify not only the amount of collagen, but more importantly, its histologic features, its association with inflammatory cells, its association with steatosis, and a lot of other relationships in the biopsy that are just beyond the scope of even the best pathologists like Dr. Behling and Kleiner to do. So we have a lot of work to do. I recognize we're confronted with the need to develop biomarkers that are predictive and that are hopefully non-invasive. But at the same time, we need to continue to dig deep to understand pathophysiology, heterogeneity, and sort of prospects for new biology around the disease and its regression.
Thank you, Scott.
And now I would like to introduce Dr. Gregory Levin. Dr. Levin is the Associate Director for Statistical Science and Policy in CDER at FDA, and he's supported drug review in several areas, including hepatology, and plays an active role in how CDER considers benefit risk in the evaluation of effectiveness. Greg, can you give your few remarks?
Sure, and thanks for the opportunity to speak to you all today. I learned a lot in the talks this morning, and mostly just want to reiterate a few of the important points that I think you heard from other speakers this morning. If there's one message that I want to make sure is kind of taken away from the sessions this morning, it's that correlation between a biomarker and a clinical endpoint in epidemiologic studies is necessary, but not sufficient to support that a biomarker is a reliable surrogate endpoint. So in other words, correlation is not sufficient to support that drug effects on the biomarker will reliably predict drug effects on the clinical endpoint. In earlier talks today, I think you heard Dr. Stein and Dr. Hager and Dr. Heller, among others, kind of walk through a variety of causal diagrams that illustrate the many ways that a biomarker can be a correlate of clinical outcomes, but not a good surrogate. And if this is the case, carrying out a clinical trial that looks only at the effects on the surrogate can lead to incorrect conclusions about the benefits and the benefit risk profile of a new drug. And unfortunately, we do have many examples of biomarkers that were strong correlates, but clinical trial evidence ultimately did not confirm that they were good surrogates. So because of the insufficiency of correlation, when we see correlation between a biomarker and a surrogate, we're ideally going to want that to be supported by an overview of data from multiple randomized clinical trials showing that drug effects on the surrogate reliably predict drug effects on the clinical endpoint, or at minimum, a sufficiently comprehensive biologic understanding about the principal pathways through which the disease process affects patient outcomes, as well as a good understanding about the mechanisms of the drug and its potential on and off target effects. And developing such a good understanding is very challenging, and it's a high bar. So moving to kind of the case that we're talking about today with non-cirrhotic NASH, there are data, and many of those epidemiologic studies were presented in the talks in session 2 this morning. There are data that shows correlation between these histology-based measures of fibrosis and steatohepatitis and long-term clinical outcomes, such as liver transplantation and death. And as you heard from Dr. Makar earlier, while acknowledging the complexity of NASH, this correlation was supported by what was considered a sufficiently good understanding of the disease causal pathway to support the use of endpoints defined by improvement in fibrosis and or resolution of steatohepatitis as reasonably likely surrogates to support an accelerated approval. However, I just really want to underscore that there are no randomized clinical trial data to support that these biomarkers are causal, not just correlates. In other words, there's no data showing that treatment effects on the surrogate will reliably predict longer-term treatment effects on the clinical outcomes. So as a result, I believe there still remains uncertainty about the nature of the relationship, the nature of the causal relationship between these biomarkers and clinical endpoints, including what magnitude and duration of effects on fibrosis and on steatohepatitis will be necessary to actually translate into clinical benefit. And notably, in other therapeutic areas, even with the best surrogate endpoints, you often need effect sizes that are quite large and are quite durable on the surrogates to translate into meaningful clinical benefit. So given this state of knowledge, I think there's going to be a lot of uncertainty around the potential extent of clinical benefit after trials to support an accelerated approval. And there might even be greater uncertainty around the benefit risk profile if there's the potential for any serious risks of a given drug. And so similar to, I think, the final slide that Dr. Heller had in the session 2 presentations, I think these uncertainties underscore why it's so important that randomized clinical trials are conducted to confirm clinical benefit and evaluate long-term benefit risk for drugs that are granted accelerated approval. So, and the last quick point I want to make is that while there are clear challenges to feasibly and reliably carry out these clinical benefit trials after an accelerated approval, I do believe that they can be successfully completed in a timely and reliable manner. And I can comment more on some steps to achieve that. I believe we may have some questions on that in the Q&A area. So I'll stop there.
Thank you, Greg. So if all of our panelists can turn on their cameras, I think everybody's on. I'm going to ask our first question that we have. And that is, there is uncertainty about the magnitude of clinical benefit of one stage of fibrosis reduction or MASH, NASH resolution on histology. What are some of the ways to describe or quantify that potential benefit? And so, Scott, I'm going to start with you.
Sure. Well, I think it's a very important question, of course, and I think that the first answer is it really depends on where the one-stage benefit is occurring. I think the FDA has indicated previously, and correct me if I'm wrong, that, for example, failure or if a drug stops progression from F3 to F4, that's a clinically meaningful benefit. So I would infer from that regression from F4 to F3 is the most meaningful one-stage improvement that one could see, whereas F2 to F1 probably has modest, if any, clinical benefit. So I think that the first answer is that the higher the fibrosis stage, the more clinical benefit will follow if there's regression. But we also understand that the fibrosis content in the liver is not linear, and the change in the amount of collagen, for example, that between F3 and F4 is about 5-fold. So that means it's going to be a higher bar or harder to regress F4 to F3 in order to achieve that clinically meaningful benefit.
Thank you. Don or others, do you have something to add in?
Yeah, I would add in, and I think this may be a recurring theme. So I would argue that given an understanding of the biology of fibrosis and the progression thereof, that it's not so important about the magnitude of change. Certainly change from F4 to F3 is clinically meaningful. I would submit that change from F3 to F2 is clinically meaningful over a longer period of time, or even F2 to F1, or eradication of fibrosis from F1 to F0 is important. It may take 10 years to see that benefit, but given that we know that unless you remove the underlying lesion, the underlying biology, fibrosis is going to progress. I would submit that maintaining a steady level of fibrosis or any resolution of fibrosis is going to be ultimately clinically meaningful. So I think, again, I made the comment that it's not so much whether resolution is important, it's how to measure this. I think, measuring the difference between, we're going to talk about this tomorrow, the difference from F2 to F1 or F3 to F2 is going to be difficult, especially non-invasively. But any resolution, in my view, is biologically important.
And just to put a fine point on that, preventing progression in a non-cirrhotic stage would be a very important and achievable endpoint. If you permit a patient, or if a treatment permits a patient to stay at F2 or F3 and never advance into the cirrhotic stage where we understand the complications are likely to ensue, that would be a very important benefit. Having said that, we'd have to still remain vigilant for the risk of hepatocellular carcinoma, particularly in F3 patients, even if the fibrosis doesn't progress.
Yeah, that's what I was trying to say. Scott just said it better.
So I don't know if anybody else wants to.
David has his arm
Yeah.
raised. David.
So I guess one question I would just throw out there for thought is, when you say you have a therapeutic intervention that over the course of a couple of years reduces the fibrosis by a stage, cirrhosis to bridging or bridging to stage 2, what have you, have you really reset the patient's disease such that now we all saw the curves that were shown in the presentations. Have you shifted the patient up to a more slowly progressive curve going from cirrhosis to F3? Or is it really a bit of an illusion in the sense that as soon as you withdraw the therapy, the natural disease in that particular patient takes over again, and they rapidly, not over 7 years, say, but they within a year or two go right back where they were before. And so what does it mean to hold something static? How is that achieved? That sort of thing. And I think that's something that we really don't know.
So there's no guidance from animal studies insofar as how quickly fibrosis will recur if the insult is resumed. But we do assume that NASH, like any chronic disease, is going to require chronic therapy. I mean, after all, we wouldn't expect the LDL to stay low if we have a patient who's hypercholesterolemic and we stop their statin. So I think we have to have the same mindset about, benefit will only accrue as long as the drug is being given and effective. We're not going to turn it off the way we can do with Hep C cures.
And I am going to ask I don't want to disrupt the conversation too much, but if you have something to add on, to please raise your hand and I'll call on folks in order unless there's something that you feel you've just got to jump in on. So Maru, I think you had something to chime in here, and then Dawn.
Yes, thank you. So just basically on what David was saying, in fact, I was going to ask the exact same question. The corollary would be with alcohol-associated liver disease, where you have that initial activation of stellate cells, then they become quiet, right? But then they're very quickly reactivated again. And so I didn't know, in fact, if there was a reason to think that should be the case in this disease. I feel like there should be. But as Scott said, there's no data that's been shown that actually demonstrates that.
And Dawn?
Yeah, I would agree both with David, Maru, and Scott. I mean, I think that we just don't know yet, right? So, there are data. The data are that stellate cells can, and Scott has shown this elegantly in lab studies, that stellate cells that become activated can become quiescent, then go back to being activated. So there is a dynamism here. I tried to emphasize some of this. And so, but we don't know what that means for the patient clinically. There is one other point I would make, and that is, remember that the lesion that is a problem clinically is clinically significant portal hypertension. And so if we can prevent that, I don't really care whether my stellate cells are activated or not, as long as they're not making enough fibrosis to lead to CSPH, I would be happy.
Okay, Scott.
So completely agree, Don. And there is evidence that once stellate cells are activated and they quiesce, their reactivation is accelerated. And that's probably regulated by epigenetic control. I have a very talented new faculty, Shuang Wang, in my lab, who's presented some of the data in abstract form, that the epigenome of stellate cells resets and the cells harbor what's called a fibrogenic memory. But we don't know yet whether that translates into accelerated recurrence of fibrosis. So there is some insight, but not enough to answer David's question.
And Theo?
I would like to extend some of the comments that have been made. I would say that it may not be as simple as fibrosis, that maybe HCC risk relates to duration of disease. Maybe development of clinically significant portal hypertension is not just fibrosis. And we see that, for example, in NASH already, there's plenty of evidence showing that the portal hypertension is out of proportion to what we'd expect with a fibrosis. So while I think regression of fibrosis is a very worthwhile aim, I would want to be sure that that's going to achieve the ends that we would like.
Okay, Scott?
Just a quick comment to amplify Theo's comments. We do know that the inflammatory microenvironment in MASH is different than in Hep C, particularly with respect to propensity to cancer and the responsiveness to checkpoint inhibitors. So it shouldn't surprise us that things other than fibrosis can also be determinant of outcomes in MASH.
Okay. Last call on this very active question. Naga, did you have anything to add?
Yeah, I was going to, before asking the second question, I was going to pose a follow-up to maybe Don and others. What's sort of the sweet spot delta between, if you're in a randomized control trial, I think this has come up at the AC meeting with obeticholic acid. What should be the delta between active treatment versus placebo? Is it, related to the delta is it related to the numbers needed to treat or the duration? How long? Can you just sort of share your thoughts, Don, or maybe Maru and Scott and others as well?
Yeah, Naga, I think that's a great question. And I think it comes down to numbers. You kind of indicated this. I think if you think back to the cardiovascular trials, right? So some of those trials included 20, 30, 40,000 patients, and they were able to show improvements in MI and outcomes of 1% because they had so many patients, and yet they led to approval of, calcium channel blockers and beta blockers and various other drugs. So I really think it's a numbers game. From a pragmatic standpoint, there's no way that we're going to see trials in NASH of, thousands of patients. So I think there needs to be a statistically significant difference of some sort. But again, I would go back to if you can show a lack of progression, because again, we know the biology of the disease and these patients are going to progress. So I would be happy with numbers that are statistically different. I think the obeticholic acid data were pretty convincing. Those were significant changes, and I think that if you could sustain those, you probably ultimately would have an effect on clinical outcomes.
Thank you, Don. Scott, and maybe I think you.
Very quick. I think expecting a full stage improvement is worthy, but maybe a high bar. And I think increasingly we recognize that if there's demonstrable improvements by AI, and we've used one of the platforms mostly in our experimental work, the clinical benefit can be significant. So I hope we can continue to validate the AI-based method so that we can determine whether subtle changes are still meaningful in terms of clinical outcomes. Some of that may be able to be gleaned from a retrospective look at samples from the ongoing or completed phase 3 trials. But for sure, I personally believe that reduction of less than one stage of fibrosis is still very important, but we just aren't capturing that using current methods.
Scott, just would you when you look at this, would you combine improvement plus lack of progression into sort of one category?
From a clinical perspective, perhaps. From a rigorous kind of validation perspective, no. I would strictly quantify the different elements of the biopsy by AI and try to identify and establish a score, again, that includes collagen content, but other features of the biopsy that can be quantified by AI.
Greg, I think you raised your hand. Sorry.
Yeah, thanks. So a couple comments. First is, as I mentioned in kind of my opening remarks, I think we have examples that have illustrated that the magnitude of the effect on the surrogate matters. In some cases, there have been surrogates where a certain magnitude and duration of an effect has been necessary to reliably translate to clinical benefit. So I think we do need to think about the magnitude of the effect and how and whether it's large enough. Also, in some settings, like for example with LDL, which was brought up earlier, and blood pressure, we have randomized clinical trial data that has allowed people to build models to translate an effect on the biomarker, say LDL, into a predicted effect on the clinical outcome, say reduction in cardiovascular risk. And those models can be used to estimate the actual magnitude of clinical benefit based on a trial that evaluates the surrogate. We do not have such data in NASH. So I think that what that ultimately leads to is uncertainty about the magnitude of clinical benefit when you see a trial evaluating the histology-based surrogates. And this becomes particularly challenging, I think, when we think about benefit risk. I think Dr. Stein mentioned earlier that we ultimately make benefit risk assessments based on the magnitude of direct patient benefit, effects on how patients function, feel, or survive. When we have a trial with a surrogate, there's going to be uncertainty about the clinical benefit, which is going to become, I think, particularly important, particularly challenging when there are potential risks of the drug. So I think, I do think that, recognizing just the uncertainty that will exist about clinical benefit and how that translates into the benefit risk assessment is important.
Thank you, Greg. Don, if you have a quick comment, because there are others who are interested to comment as well.
Yes, Naga, just very quickly, and I just wanted to come back to your comment, which I think is very important, or your question, which is very important, and that is about the difference in fibrosis versus inflammation. And this comes back to mechanisms. So remember, the mechanisms of fibrosis and inflammation are probably different, and therefore we probably are going to need in this field dual or triple therapy. So a fibrosis target is a drug that targets that versus a drug that targets the underlying disease or inflammation. And so I hopefully we'll get into that a little bit further, but I think that's going to be a very important point, that it's not going to be, if you have one drug that does everything, great, but I think that's unlikely. I think we're looking at two or three different mechanisms and 2 or 3 different drugs.
Thank you. A great point, but let's just stay focused on endpoints and clinical trials. Dr. Bailey.
So I wanted to comment on the fibrosis issue because there are 2 things about fibrosis actually that can impact its physiologic effect in the liver. One is the location of the fibrosis, and the second is the quantity of the fibrosis. And so, I think both are actually important in how the disease is changing the architecture of the liver and therefore changing the function of the liver. And I think it's really important to just recognize that there are these 2 components. So you could have a patient who has a lot of fibrosis. It's all limited to zone 3. It's extensive. and it would be staged as a 1B, although quantity-wise, there may be a lot of fibrosis there. You can also have a patient who has the same amount of collagen deposition, but it's in the portal tracts and the central veins, and it has formed bridges. And that patient may have a different clinical course, a different progression rate, and that fibrosis might have a different effect on how the liver functions. So I just think from a pathology point of view, it's important to understand there's two components to fibrosis.
Thank you. Maru, I think you had your hand up.
Yeah, thanks, Naga. A lot of what I was going to say is sort of been said, but I do want to emphasize that we need to really focus on leveraging the data that we already have to see if we can get our hands around what would be a clinically meaningful change in fibrosis quantity, and whether it has to do with geographic location or quantity, or a combination of the two through AI-based methods. I think we really need to do that, because when we are in the F3 population where we have such a broad range of fibrosis burden, it's really important for us to understand when a therapeutic is having a significant impact, if it halves the amount of fibrosis, but you're still, in stage 3. I do think that's really meaningful, and we need to find a way to illustrate that.
Thank you. Thank you. I'm going to move on to actually question 2. Great discussion. I think this has come up at the most recent AC meeting. If a drug has an anti-fibrotic effect with fibrosis reduction, but underlying necroinflammation or disease activity were to remain the same, what is the clinical meaningfulness of this particular phenomena? And then as a follow-up to that, for example, I think EMA requires an endpoint to have both NASH resolution as well as fibrosis improvement. Combine both, that's not the case with the guidance from the FDA. Maybe Maru, you can start, and I think this is really important. Maybe all of you can chime in on this particular one. Maru?
Yeah, thank you. So I guess the way I look at this, it's sort of like putting the brakes on without taking your foot off the gas, which doesn't seem like an intuitively smart thing to do. And if you think about it in the sense of viral hepatitis, right? Nobody would ever say, well, we should just treat hepatitis C with an antifibrotic, right? So you definitely need to treat the driver of the disease. And so I firmly believe that you have to improve steatohepatitis to have a real treatment for this disease. Now, that being said, it's possible that steatohepatitis may lag for whatever reason. And so I could imagine that there could be a situation where you could have fibrosis improvement in isolation, but you have other surrogates that are moving in the right direction, because what you don't want to do is improve fibrosis, but not get at the underlying mechanism of disease. And then you don't know what that's going to do to cardiovascular disease, vascular inflammation, carcinogenesis, et cetera. So I guess and then the last question was regarding EMA, approach, which would be a dual. I don't know if I want to go so far as to say that, but I do think that we need to make sure we understand the importance of disease driver and the disease.
Thank you, Maru. Scott, can you comment on the first part of what Maru had said about improvement in fibrosis alone without improvement in necroinflammation may not be, you were the PI for the Senicriviroc, where it was primarily antifibrotic. So, and then after you, maybe Don could comment in as well. I'll come to you, David.
Sure, I completely agree with Maru. It's improving fibrosis is necessary, but not sufficient. Because clearly, and we don't have the benefit of what antifibrotic responses look like, neither in liver and really not so much in any organ either. There are only 2 drugs approved for pulmonary fibrosis, and they only slow deterioration. So we have a chronic inflammatory disease. We have to acknowledge that fibrosis is a consequence of that. And so addressing fibrosis solely from a therapeutic perspective will probably be inadequate, even if you believe that fibrosis begets more inflammation. I think both a primary disease a target that attenuates the primary disease and one that attenuates fibrosis are both needed. If it's in the same drug, more the better.
Scott, though, is it inadequate from a biology standpoint? What about, is it that fibrosis alone without improvement in steatohepatitis, is it inadequate as an endpoint, as a surrogate clinical trial endpoint?
Well, as you know, we don't have that data, but I would argue that's meaningful. It's just not optimal. In other words, since we all agree that the most dreaded complications of advanced MASH are, in fact, complications of portal hypertension and perhaps cancer, I think it's reasonable to expect that if we attenuate fibrosis, we will move the patient further away from developing those end-stage complications, but it will be swimming upstream in the face of ongoing inflammation and injury wrought by the underlying disease.
Don, and then David.
Yeah, it's a very intriguing question. I agree with Scott Maru's comments. It harkens back to an experiment that was done a long time ago. So if you overexpress interferon gamma in a mouse liver, in fact, there was a transgenic mouse where this happened, you get an incredibly remarkable inflammatory lesion with no fibrosis because interferon gamma is so antifibrotic. Now, I don't know whether those mice develop portal hypertension or clinical, what their clinical features are, but I don't think we really know what the clinical outcome would be of a situation where you had ongoing inflammation and you had stalled fibrosis. I just don't know. Ideally, again, this is where I come back to the comment I made before. We're going to need to target both of these probably. But
I would think that such patients with progressive NASH but without fibrosis may be susceptible to like drug toxicity, just because mitochondria are vulnerable and so forth.
I'm sure it's not a good thing, but I don't know in terms of the clinical phenotyping. I just don't think we really know.
David, thanks for your patience, by the way.
No problem. It's all very interesting. So, from a histologic standpoint, it's hard for me to imagine what this might look like, just because of the close association with, especially ballooning and inflammation, with the appearance of fibrosis in the sinusoids and that sort of thing. So it's a little bit difficult for me to imagine what this might look like, and maybe that's why, it's been difficult to demonstrate efficacy in some of these situations where antifibrotics have been used alone, even when other tools have been used, AI and quantification of collagen. One thing about sort of the evaluation, though, and both of these things being important, I think that the only situation where you get reliable in the sense of, I'm sure this is real, and clinically meaningful benefit is when both of those things change. So, the data that I showed is that when the activity decreases, those are the patients who actually improve their fibrosis. And if you just, on a numbers standpoint, saw a stage change without, say, comparing the biopsies side by side, but nothing else changed, or maybe the activity actually went up, I would worry that really was noise, and that you aren't looking at a really true fibrotic response, that you're just looking at sample variability or whatever. So having both of those things go in the same direction, to me, makes that a much more meaningful histologic change.
Okay, great. Thank you, David. Laura, I think maybe time for next question.
Sure. So our next question would be, what is the clinical meaningfulness in the reduction of liver steatosis? So published data, we're in a theme here. Published data are not consistent.
You're breaking up. Did anybody hear the rest of this?
Now you're muted.
Okay, my Zoom keeps killing here, so I will try again. So what is the clinical meaningfulness and the reduction of liver steatosis? Published data are not consistent in supporting the reduction of fat as clinically meaningful, and with progression of fibrosis to cirrhosis, the reduction of fat is observed on histology. Now, I know several folks have been talking around this quite a bit. So I don't know if David, you want to kick us off, you or Cindy?
Okay. So, I think that the fact when we see improvement in the other features in the natural history study, or at least in the clinical trials that I've been familiar with, the fat does go with it. So the fat will improve along with reduction of inflammation and ballooning. And so that by adding it into the score, you get kind of a magnification of the effect. As a single feature, though, everything else unchanged, I don't think that that's necessarily meaningful. I can say that we did look at that feature. It didn't get into the paper, but we did look at steatosis alone in the large natural history study. And the patients who got better, who regressed over the time period, did actually drop their steatosis. But I think as a single biomarker, measuring either, imaging the biopsy or by MRI, PDFF, or some other, non-invasive test, I think it's hard because of the sort of relationship of steatosis degree with fibrosis overall to know where you are on that curve. I think you would have to be very careful about how you interpreted just a decrease in steatosis. And so I'm not sure that it has all that much meaning on its own.
So I see that Don has his hand raised, but I also wasn't sure if Cindy, you had quite a bit of discussion on this during your presentation, if you wanted to comment now, or do you want to go after Don?
I'll go after Don. Go ahead.
Well, I actually was just going to ask David and the pathology crowd about the clinical observation, and we see this in the clinic. Patients who have advanced fibrosis and cirrhosis, even though they continue to have the metabolic syndrome, the disease hasn't gone away, they kind of get what we call burnout steatosis. And I don't know what that means. And so I was going to ask you guys, do you agree with that, or do you think that is clinically a clue as to what's going on here?
Well, a lot of things change as the fibrosis gets to cirrhosis. There's a lot of blood flow changes through the liver, and so nutrient delivery and the way that the liver can handle those things changes as you progress to end stage. And that may have, effects on how you store fat and maybe some of these other things as well. I mean, I don't see a lot of cirrhotics because they tend not to be chosen for clinical trials and get biopsied and everything like that. But a fair number of them still have very active disease with a lot of fat. And yes, it's true that some burn out, but there are others that are still quite active. And some of the most active cases that I've seen have either, close to cirrhosis or cirrhosis on biopsy.
I would agree with that, David, that I guess it's simplistic and maybe just purely sort of morphologic, but I think that once you get very, very advanced fibrosis with a lot of collagen in the liver, you are in fact altering the blood supply in a way that changes what those hepatocytes are seeing that's coming in and how they're metabolizing it. And I think that might be part of the reason why we see that reduction of fat. But I do also agree that I would say the majority of cirrhotics, at least with the clinical presentation consistent with fatty liver disease, still do have some residual steatosis and inflammatory activity.
Okay, and Scott?
Just a brief comment and to introduce a concept that I think addresses some of the heterogeneity, and that is that we should bear in mind, at least as far as fibrosis is concerned, that the repertoire of therapeutic targets for fibrosis are likely to evolve as the disease advances, such that at the end stage, when there's less fat, there are more autocrine pathways that stellate cells are engaged more with each other and with endothelial cells than with inflammatory cells. And so it may well be that a nanofibrotic that works in F2 or F3 may not work as well in F4. We showed that in a paper that had both human and murine data earlier this year in Science Translational Medicine. But I think the concept is this is a moving target, and we need to be mindful of that.
Any other comments before we move on? Okay, I think you have the next question.
Thank you. Thank you. Really wonderful discussion. Once again, though, I just remind keep your thinking, the clinical trials endpoints as sort of the focus for your thinking and how for your comments. So the next question, I think both Don and Scott, you have touched during your comments. What is the clinical benefit of reduction in fibrosis from, let's say, stage 4 to stage 3 versus stage 3 to stage 2? Once again, this may just be the function of duration, but as the clinical trials are designed, as the agency looks at these programs, sort of what are your perspectives? Maybe start with, I'll actually ask Theo, followed by Don and Scott. Theo?
You're muted, Theo.
How about now? Thank you. I think this is a really interesting question. And I think I agree completely with what Scott said about not being misled by viral hepatitis. But in viral hepatitis and David's published work in JAMA Open, there are patients who get worse and there are patients who get better. And in the best viral hepatitis studies, 50% of patients improve. So we have to talk about when we talk about a one-stage improvement across that sort of data, I don't know how we're going to get that kind of resolution on a population level. For an individual patient, it would be very nice. If you ask me for an individual patient going from F4 to F3, I agree with what Don and Scott said before, that would be very helpful. I think that would be very important, with the caveat that we don't understand if that really translates completely to clinical benefit. We know that worsening translates to clinical adverse outcome, but the converse has not yet been proven. So I worry about these sort of endpoints for population, and I worry about splitting down too fine. As Maru said, we don't have a perfect system. So the problem is we're going to throw out histology, but we don't have anything better that's validated in a better way. So we have to be careful. We have to be careful what we ask for, because if we throw out what we have, we may end up with worse. Long answer. The simple answer would be yes. I think any decrease in histology will be something that we should aim for, and then subsequent validation should be sought.
Don, then Scott.
Yeah, I mean, similar to what we said before, I'm all in for any reduction or any a holding pattern, I think is going to be good clinically. Just one other idea and one other thought that is very important in designing clinical trials and thinking about this whole field is that what we don't know, the holy grail here is we don't know which patients are going to respond and which ones are not. So it's very clear that some patients are going to have a reduction in fibrosis. We've seen this with hepatitis B and hepatitis C, for example, but other patients are not going to respond. So figuring out which of the patients are going to respond is really important. And we don't have any great answers right now. We need more data.
Scott.
Little to add to the two previous discussions, but I would mention another concept that we haven't touched on, and that is the point of no return. And here I would harken back to the work that David cited about Ian Wanless, because I think he turned out to be very prescient in his prediction that once you start to get vascular extinction and distortion of blood flow in the cirrhotic liver, you may improve fibrosis and even metabolic function, but you're probably not going to restore or reverse fibrosis or cirrhosis completely. And so that's a stage too far, so to speak, in terms of where we engage patients in clinical trials.
Thank you. I think we will go on to the next question, Laura Lee.
Sure. So given that there are multiple competing phase 4 confirmatory trials that may be ongoing with blinded placebo arms, if multiple agents are approved via the accelerated path, how can this challenge for industry stakeholders be addressed? And so a few other questions that may come along with this, kind of what are some of your suggestions to ensure that clinical benefit trials will be completed without high rates of dropout and missing data? And how can we encourage the sites and patients to remain in a clinical benefit trial if a product or products are approved via the accelerated approval pathway, or if the product's not approved under accelerated approval, but the clinical benefit trial is ongoing? So I'm going to turn first to Greg, because I figured, well, we're here today to talk about NASH and NITs. You also have kind of that bigger picture view that maybe would be useful for us here.
Sure, thanks. This is a really important question, and it's a big challenge. So definitely want to underscore that this is a big challenge. But I kind of took some notes. I had 5 things that I think could help in this manner. The first is ensuring that the clinical benefit trials are well underway at the time of the submission of a marketing application for an accelerated approval. Ideally, being fully enrolled such that the accelerated approval decision would not impact enrollment. The second thing is kind of just having a really careful and appropriate consent process of participants that enter the trial that openly discusses the uncertainty around the surrogate endpoint and the uncertainty about the benefit risk profile in the event of an accelerated approval, and the importance to public health and to patients with NASH of understanding whether there's clinical benefit, so that patients know that it's important to stay in the trial after an accelerated approval to answer the questions that the trial was intended to address. The third thing is just kind of following steps that have been recommended in many settings for preventing missing data, like discontinuation, clearly distinguishing between treatment discontinuation and study withdrawal, having a careful definition of withdrawal of consent so that it actually means that a patient withdraws their consent to contribute additional outcome information. It's not used to mean that a patient doesn't want to receive treatment anymore. So just, training of investigators on the importance of retaining patients in the trial, things like that would apply regardless of the setting. The fourth thing is ensuring that the interim clinical endpoint results, if it's a seamless phase 3-4 trial, which was an option that was discussed, that those interim clinical endpoint results are kept confidential, that there's maintaining confidentiality of interim results during an ongoing trial is an important principle of good conduct. And so trying to make sure that happens. And then the last thing, which is, I think, a little bit unique or hasn't really been carried out in this setting, but I think could really help, is the implementation of performance standards for important clinical trial conduct aspects, such as enrollment, adherence to treatment, retention. So performance standards documents have been used in other post-marketing settings, like post-marketing safety trials. And what these do is they include planned measures to support each aspect of trial conduct. They set target levels and minimally acceptable levels, and they document actions that will be taken if the trials aren't meeting those levels, and approaches that will be used to monitor performance. I think they've helped in post-marketing safety trials, and I think they could have similar value in the context of a post-marketing trial to verify clinical benefit.
Okay, Rocky, I see you have your hand up.
Yeah, I don't really have any silver bullet suggestions other than what Greg suggested, but I'll tell you, this is an incredibly important question, and we are seeing this in real life now. So this is particularly true with some of the new agents, the GLP-1s in particular. Patients want to be on those. There is a lot of lay press out there, and this is going to be a huge challenge for the field. So I'm really curious to hear what the clinical trialists think about how this is going to be dealt with.
And that might be a follow-up that I would ask Greg as well. And I don't know if others have thoughts, especially when you've got an Excel how are you going to continue to maintain that confidentiality?
Well, so in the event of an accelerated approval, I mean, certainly the results on the surrogate endpoint that were used to support the accelerated approval are going to be disseminated to the public. I think that is the nature of this kind of approval pathway. But if it's a seamless, phase 3, phase 4 trial, I think having confidentiality of the interim clinical endpoint results, so interim results on the composite clinical endpoint that will ultimately be used to verify clinical benefit. I think that can be achieved. And, I think analogous to, for example, like the type 2 diabetes trials that were carried out, where there were post-marketing continuation of the trials to confirm the cardiovascular safety of the products, and they maintain the confidentiality of interim cardiovascular outcome data. I think similar approaches could be taken here to ensure that the conduct maintained maintained appropriate in a post-marketing setting, along with some of the other steps. I think that's only one of many, but I think that can be achieved.
Okay, so I'm going to go Theo and then Maru.
I'll point out that this is a problem in many respects, and it's not a new problem. For example, hepatitis C, once the first drugs came out, there were other studies that continued, but they did recruit and they did accrue and they did finish. And I think it's a problem in hypertension. It's a problem in diabetes, but people want to be part of research. And I think in an appropriate setting, people will stay in studies if there is a process of informed consent and open discussion. Not to minimize the problem, but I think it's been done before and I think it will be done again. It just has to be done correctly.
Thank you. Maru?
Yeah, just more comments. So to Greg's comments. So these are really, tenets of, trial, integrity and the way trials should be conducted anyway, right? But my concern is that it's going to be very difficult to make the argument on a large scale that, for example, somebody with diabetes not be put on a GLP-1 or an SGLT2 inhibitor because there's cardiovascular benefit that's been proven in those drugs, right? So is there a way beyond doing the usual things that you mentioned that we can, are there statistical ways to manage people who on trial will convert over onto, say, a GLP-1, for example, because it's everywhere. Prescriptions are increasing, in a logarithmic manner for those drugs in particular. So how are we going to manage that? Because it's going to be the reality.
Really, can I make a comment?
Oh, sure, Naga, go ahead.
Yeah, I just want to, as Don, you said, I don't have a magic answer here either. But I'd like to make a couple of comments, though. Just because a drug is approved doesn't mean it is standard of care. That's one. Okay. The second point I'd like to make is this is a slowly progressive disease. And it doesn't mean that there isn't a window for somebody to be in a placebo and then a crossover. So I hope that we, whether it's the GLP-1 agonist or a drug gets approved, and I think for a period of time, I hope we retain the ability to do rigorous placebo-controlled trials.
And so, Greg, did you want to comment? If not, I have a few things I could say as well.
I guess the only thing I would say is, I think if I understood it correctly, I think there were points being brought up about concomitant medications, which I think is an important discussion about what background standard of care can or should be allowed in these trials. But as I guess I could just distinguish that a little bit between crossover between arms and dropout from the trial, which are things that I think can be mitigated. As Dr. Heller pointed out, these have been we have other therapeutic areas where such post-marketing trials have been carried out and reliably completed. So I think they can, but I don't have much more to add on that.
And as somebody who's been in multiple different parts of both conducting research and now as a regulator, I do think that it's really important as you are developing trials and as you're adjusting trials along the way. But I think in this case, it's very clear that we could prospectively be planning, realizing that products could be coming on the market. So what types of interim analyses might need to be there? What types of flexibilities and adaptive design should be built into trials? And sometimes products can be added on. So to the point of like, if people are going to be dropping in or dropping out, and if they are going to be taking additional concomitant medications, thinking about how that might move together. But one thing that many times is missing, and I think is incredibly important, is there can be safety issues of putting these drugs on top of each other, and also long-term safety issues. And as somebody who's had to be in the room as people were being consented and reconsented and trying to figure out how do we write letters to the people that are in our trials as we're considering, what do we say about what is now in the popular press? A lot of times we have to really talk about that uncertainty and about how important it is to keep going to that clinical benefit. Because, and as a regulator, I'll say, like, we have to have data to make a decision, right? And a lot of times we jump on whatever we think sounds positive as the consumer of health. My family emails me all the time about things like that. And we do, as we're talking and as we're continually talking, because consent is a process that goes throughout the entire trial. It's not what you do at the very beginning. I think it's very important that we emphasize the need to have the outcomes, the issues that have come up in other situations, some of which we heard about earlier today, to make sure that all of our patients in the trials understand why we're asking them to continue in the trial, and we're not putting everybody on a given treatment, even though it may have accelerated approval, for example. Because some people may say, like, hey, this is approved, so is it ethical not to give it to them? But having seen a lot of things go very badly in my professional career, I would say it's incredibly ethical. You may really wish that you had stayed on that placebo. So I do think that we need to be very careful, and it's a hard conversation. It is a hard conversation. I'm not diminishing that, especially to all the MDs I'm sitting here with who have to have those conversations. But it is something to think about, and I think that maybe all of us can take in thinking about our engagement with patients and how we do our patient-focused development. So are there any other comments on this question before we move on? Okay, hearing none. I didn't mean to make everyone quiet. Naga, did you have any other comments? Yeah. Or
I don't have any comments, but I could bring up the next question, Laura-Lee. I think we are like the last 7 minutes or 8 minutes, a really wonderful panel, great discussion. I'm sure audience are appreciating your thoughtful responses. Next question, though, is maybe to the pathologist and the clinical trialist. NASH resolution. Is there a way to simplify documentation of NASH resolution in a clinical trial setting? I know everybody, every meeting we go, people bring up about ballooning, how hard it is. Maybe David and Cynthia and others.
NASH resolution. Yeah. To me, to be honest, and I've expressed this at other meetings, to me, this is a disease like diabetes or hypertension or something. If I take hypertensive medication because my blood pressure high and I go into the doctor's office and it's normal, I still have hypertension, but I'm on a medication, right? So the disease is suppressed, but it hasn't really resolved. So I think that, I think it's reasonable to talk about activity suppression as a way to, not really get around the problem, but at least document that you're affecting biological processes that lead to steatosis, inflammation, and ballooning. And those effects may not be equal, right? You may affect one arm or the other, and that's an important thing to recognize. But I think that, you know, when we first devised this construct, this scoring system, that was what the pathologists were kind of thinking of, you know, is can we come up with a measure that reflects suppression of disease activity? And I think even though we had, you know, we were guessing at the time what might be important, actually those things probably are important to different degrees. So ballooning is important. I think we've demonstrated that. Inflammation is maybe a little less important, at least lobular inflammation. Maybe we should be paying more attention to what's in the portal area. And steatosis is maybe the least of the three, even though it gets more points. But still, I think when you address the disease, you drop all of those things and the activity goes down and then the fibrosis improves. And so, you know, to me, that's suppression of the disease, and similarly to the hypertensive thing, you're suppressing the feature, right? That causes.
What is that formula would look like?
Well, you want something, you know, there have been any number of clinical trials that have used disease activity suppression as an output, I mean, or an endpoint, right? We did that with Pivens and, you know, I mean, what you want is something that gives you the idea that the drug is doing what it should do, right? I mean, all of these are kind of surrogates for the feels, function, survives thing. You want to know that the drug actually is affecting the disease in the liver, and that's disease activity, and then fibrosis will follow, and it always takes longer to resolve the fibrosis. But you know, 2-point improvement, you can talk about a 3-point improvement, something like that. You know, that I think reflects what's going on.
Thank you, David. And I think we just down to last 2 minutes. Dr. Behling, maybe you could add.
Sure. I think that I think the question is the number. When you compare screening biopsies and treated biopsies, it's pretty easy for people to say whether the treatment had a positive effect on that biologic process. It's better. I think the question is, how much better does it need to get before we say that's good enough? And I think, you know, if you look at the published trials, there's a subset of patients that meet all the criteria we certainly have. And then there's another subset of patients who all are trending in the right direction. So something got better, their NAS improves, but they don't meet that 2-point current criteria. And I think that's a group where, you know, there is a positive biologic effect. Are we measuring it? And I think that's, you know, but I completely agree with David that, you know, we are improving the process. We're making the liver better, but it's not, we're not curing that.
Maybe last minute or so, I think when I polled my colleague hepatologists about NASH resolution, many sort of wondered if agency would consider going back to or considering the Pivens endpoint. And I think it was the same thing for, you know, Tonic and Flint, and that's consideration for the FDA. But seems like if you want to simplify NASH resolution, that composite could be one way to consider, is what I'm hearing. But I think that's last minute or two. If Laura Lee, you want to make any concluding remarks?
I just want to thank everybody for their participation today. And we will have an hour lunch. So thank you for everybody. I know that it's been a jam-packed morning. And I think that's all.
Thank you all.